Hello, a very good morning to everyone. I, Dr. Preeti Kaur Solanki from Faculty of Pharmacy, take this privilege to warmly welcome you all. Your presence enriches our gathering. This is the session of valedictory day of 3rd International Conference of Integrative Chemistry, Biology and Translational Medicine. The core of invited lecture is sugar modified nucleoside analogues with anti-cancer, antiviral and antimicrobial activity. Now, I would like to invite the chairperson, Professor Lasker Julian, to chair the session. I would like to invite Professor Aniko Bobus for plenary lecture. So, Professor Lasker holds a distinguished position as a professor MIT, SMART, NTU, Singapore. He also serves as a director of the Nanyang Institute of Structural Biology, where he also fulfills the role of interim director prior to establishing his laboratory at NTU in 2002. He conducted postdoctoral research at Washington University School of Medicine and Institute Pasteur. With a rich background spanning over 25 years in X-ray crystallography, Associate Professor Lasker leads a multidisciplinary laboratory focused on structural microbiology and drug design. Now, I would like to request our chairperson to lead the plenary lecture.
Okay. Yes? No? Hello. Okay, a very good morning to all. So it's a thank you for the introduction. But today is a very great pleasure to have with us Professor Aniko Borbas. So she's heading the Department of Pharmaceutical Chemistry at University of Debrecen in Hungary, and she's a very well-noted chemist. And so she has made many contributions in the field of pharmaceutical chemistry with many publications, over 200 articles she published in international journals, and gathering several thousands of citations, which underscore the impact of her work had in the last years. Yeah. So without further ado, I think let's give a very warm round of applause to welcome Professor Aniko Borbas. Thank you.
Professor Leskar, thank you very much for the kind introduction. So thank you very much for this kind introduction. Before starting my scientific presentation, I have to say thanks to the organizing committee, and a special thanks to Professor Brijesh Ratti for his generosity to invite me and my colleagues. It is a great honor to present you our new results. And you know the new name of India, Bharat, is a word which is very familiar to Hungarian ears because we have a word which we pronounce Bharat. It means friend. And anyway, it is only a coincidence, but I think it represents very well to our attitude and relationship to Indian people. It does not work. Yeah, okay. Everyone knows here that yeah. It works. Okay. Nucleoside and nucleotide and nucleic acids play a crucial role in the life of every single organisms. DNA or RNA store genetic informations, but nucleoside derivatives play a very crucial role in many biological processes as storing molecules of energy, cofactors of many enzymes, and nucleosides also play a role in biological glycosylations as substrate of glycosyl transferases. Nucleoside derivatives are also well-known drug compounds, in the field of antiviral and anti-tumor therapy, there are many nucleoside-based drugs, and nucleosides and nucleotides also used as drugs in gene silencing therapy. And the most recent achievement in the field of nucleoside-based drugs was the development of vaccines against COVID-19. In all cases, these drugs contain some chemical modifications. These modifications are made for several purposes. These modifications increase the stability, in vivo stability of the compounds, increase the cell penetrating ability, and so on. So these modifications are essential. The drugs are synthetic derivatives of native nucleosides. I show you some nucleoside drugs which contain modification, for example, on the furanose unit. Several nucleosides contain configurationally altered sugar unit, not ribose, but arabinose, for example, you can see here. And not only in the field of anti-cancer therapy, but in the field of antiviral therapy, the nucleoside drugs contain modification. For example, some truncated sugar unit can be found in many antiviral compounds in an open chain form. You can see sugar and base modified nucleoside here too. These compounds also configurationally altered sugars, not D, but R configured sugars as a furanose part. And it can be seen the broad spectrum nucleoside analog such as ribavirin and some nucleoside derivatives developed for the treatment of COVID-19. This structure containing not furanose ring but morpholine ring instead of the furanose are very efficient drugs. The phosphorodiamidate morpholine oligomers, PMOs, are very excellent drugs for the treatment of some genetic disorders. For example, it is used for the treatment of Duchenne muscular dystrophy. So, although there are many nucleoside drugs are available for the treatment, but there are some points which means that development of novel and novel nucleoside derivatives are essential. These points are the spread of resistance, emergence of new viruses. It was well demonstrated by the emergence of SARS-CoV-2 virus, which caused a global, highly deadly pandemic. So, what is our contribution to this field? We perform very basic research in this field, aiming to develop completely new nucleoside structures which can serve for further drug development. So our aim is not to target definite diseases or definite molecular targets, but develop efficient new synthetic methods and provides a large number of genuinely new chemical structures for further drug development. So it is mainly basic synthetic organic chemistry. Today I will speak about our methods and the develop new structures. One of this method is the addition of thiols to nucleoside alkenes, and the other methods is the replacement of the furanose ring in the nucleosides by nitrogen-containing heterocycles, similar to the well-known, well-developed morpholinos. We also performed some biological tests, but the main focus of our presentation will be on the synthesis. We envisioned the synthesis of a broad variety of configurationally altered nucleoside derivatives by exploiting the well-known radical-mediated thiol-ene coupling reaction. It is well known in organic synthesis, but it was never used before on nucleosides. We are very familiar with this reaction. This is a radical addition reaction. The first step is the generation of a thiol radical from a thiol, which then adds to the less substituted carbon of the carbon terminal bond, resulting in a carbon-centered radical, which then abstract a hydrogen radical from a thiol, providing the product with full regioselectivity and generating a new thiol radical. The main characteristics of this reaction is the reversibility of these steps and the electrophilic nature of the thiol radical. It is an old reaction, but we saw a renaissance of this reaction recently when the generation of the thiol radical was initiated by UV irradiation in the presence of so-called cleavable photoinitiator. This is a very efficient reaction, and we studied this reaction on not terminal double bond, but endocyclic double bonds, and we gathered many information on this reaction, and we found that performing the reaction on endo or exocyclic double bond, when a new chiral center is generated, performing the reaction at low temperature is essential to achieve high stereoselectivity. Having this method and information in hand, we started the study this reaction on nucleosides. Of course, nucleosides does not contain double bond suitable for thiol-ene coupling reaction. So our first task was to introduce double bond in given positions of the furanose unit. During this transformation, which were made by well-known synthetic methods, means the elimination of one chiral center from the compound, and by performing the thiol-ene reaction, a new chiral center is generated, and we optimized these thiol-ene reactions, radical-mediated thiol-ene reactions, and we found that performing the reaction at low temperature, typically between -80 to to minus 20 degrees centigrade, this reaction proceeds efficiently and with good to very high stereoselectivity, providing the altered, alternately or inversely configured furanose derivatives almost in all cases. I don't want to boring you with the synthetic details. I only want to show you only some selected examples. The first examples are from the case when double bond was at position 2 of the furanose ring. We optimized the reaction and found that almost all cases the reactions proceeded with D-arabino selectivity instead of the natural D-ribo, providing these derivatives with very good yield and good to very high stereoselectivity. But interestingly, when the thiol was 1 thiosugars, the stereoselectivity was very low at room temperature, and there were no stereoselectivity at these temperatures, but cooling the reaction mixture somehow increase the stereoselectivity, but only moderate stereoselectivity could be achieved even in very low temperature. The previous reactions were performed on on pyrimidine nucleosides. When we performed the same reactions on purine nucleosides, for example, adenosine derivatives, we found that the stereoselectivity was a bit lower, but higher than in the sugar case in pyrimidine nucleosides, and in this case the thiosugar and other thiols, alkyl thiols, thioacids gave the same stereochemical outcome of the reaction. These compounds were tested for their potential antiviral and anti-cancer activity. This was only simple routine testing, and we found some active compounds with good and selective activity on some tumor cell lines, and some of the compounds showed good activity against SARS-CoV virus with moderate selectivity. We also performed similar thiolene reactions at the double bond having at position 3 of the furanose unit. In this case, independently of the thiol and independently of the nucleobase, we observed very high D-xylo stereoselectivity. So all compounds were xylo configured, all products were xylo configured compounds, and this stereoselectivity was generally very high or almost exclusive, and the yields were also good. In this case, we also tested the antiviral activity of the compounds against SARS coronavirus, and we identified some promising compounds as potential antiviral agents. In this case, we also performed deprotection. Interestingly, neither of the deprotected xyloconfigured compounds were active against coronavirus, but introducing some lipophilic group at the primary position of the sugar unit led to regaining activity. Our next work directed to the replacing of the furanose unit with a nitrogen-containing heterocycle or a heterocyclic system. The most well-known compounds with the nitrogen heterocycles instead of the furanose ring is the morpholino nucleoside, but there are many other nucleoside derivatives, synthetic derivatives, such as log nucleoside, which contains nitrogen heterocycle or iminosugar instead of the native furanose unit, and there are some interesting nitrogen-containing condensed and bridged heterocycles in the nature. So these structures what inspired us to perform this synthesis. The starting point of this research was always this cyclodialdehyde obtained from oxidative cleavage of this diol part of the furanose unit, and then we performed different cyclization reactions using different amines. And these different transformations provided interesting new tricyclic, bicyclic heterocycles and also some new morpholin-containing derivatives. Our first results in this field was the synthesis of this kind of tricyclic compounds. We named this compound tricyclano in analogy of morpholino, which contain morpholino, and these compounds contain morpholin containing tricycle instead of the furanose unit. In this reaction, which is a simple reaction for performing, but mechanistically is a very complicated reaction. Several cyclization reaction occurs, and between amine and the oxo functional groups, resulting is this tricycle, and theoretically 8 possible stereoisomers can be formed in this reaction, but because 3 new chiral centers are formed in the reaction, but we observed the formation of this stereotriade in a completely stereoselective way. In all cases, only one compound was formed. It was a bit difficult to determine the absolute stereochemistry of this structure, but finally we could crystallize one of our product, and we could determine the configuration of all 3 new chiral centers. The high stereoselectivity was due to the control of the chiral centers of the starting compound. Next, we synthesized some new morpholino derivatives. In phosphordiamidate morpholinos. The morpholino monomers are coupled through phosphordiamidate bonds. We envision to couple the morpholine units directly without any bridge, any bridging element. We converted the hydroxyl function of a nucleoside into an amine and react this compound with the cyclodialdehyde to providing oligomers without any bridging element, and we hypothesized that this compound having basic character due to this tertiary amine functions have good cell penetrating ability, and indeed we could demonstrate by coupling a dinucleotide analog with body P that this compound could penetrate into the cells. Then in the frame of a collaboration with Brijesh Ratti, we studied these new nucleoside derivatives, both the thiosubstituted nucleosides and morpholine ring containing nucleosides against malaria. The preliminary tests were performed against chloroquine sensitive Plasmodium, and among the morpholine containing compounds we could identify some compounds with very high in vitro activity. Some of these compounds, however, showed toxicity to human red blood cells, and for example, the tricyclano also showed activity, but not as high as the morpholine nucleoside hybrid compounds. The nucleoside analogues obtained by thiolene reaction were also tested, and among the tested derivatives we identified 4 compounds which showed very high activity against the chloroquine sensitive Plasmodium, and then the hit compounds, these 5 hit compounds were tested against chloroquine resistant Plasmodium 2, and all compounds showed very potent activity against Plasmodium strains. The compounds have very diverse structures, so we could not really define structure activity relationships, but probably by further modifications producing more and more compounds, we could also gather some structure activity relationship information in the future. Nevertheless, the best compound was involved in an animal study, and this compound showed good activity against malaria in a mouse model. These results were published last year in a joint paper with Professor Ratti's group in Scientific Reports. Just some words on our new results. We also performed dominoes tricyclization reactions using propan triamine reagent, producing these kind of tricyclic nucleoside analogues, which we called triazide tricyclano. We also perform the deprotection, all of our compounds. These reactions also proceeded with full stereoselectivity, and these are new compounds. The results were published last year in OBC, and the compounds has not been tested in any biological tests. I have mentioned that morpholinos are well-known and well-studied nucleoside analogues, and I also showed you in the introduction that many therapeutic nucleoside derivatives contain fluorine at either the base or on the sugar unit. Quite interestingly, fluorine-containing morpholinos were not known in from the literature. We decided to combine fluorine and morpholine skeleton and performed the cyclization reaction of the starting nucleoside aldehyde with fluorine-containing amines to produce morpholino skeleton having fluorinated alkyl chain attached to the nitrogen, and we also developed a synthetic method to directly couple triflumethyl group to the nitrogen of the morpholine ring. We produced many new compounds starting from uridine, cytidine, adenosine, and also guanosine. It is not shown. And these compounds have not been tested yet, but I think these structures can be interesting, for example, testing against malaria too. Just one slide with new structures. We also perform not only cyclization or cyclization with amine under reductive conditions to produce morpholine ring containing nucleosides, but we also perform the multi-component reaction with this dialdehyde. It is a kind of Robinson-Schöpf synthesis, and using this three-component reaction, we could provide this bridged bicyclic nucleoside analog, which we called oxagranatano, because this skeleton is similar to the granatane skeleton. And we also performed further synthetic modification of this compound when we performed the reaction with allylamine. This is very new compounds. These results have not been published yet. As a conclusion, so our original aim was to develop new synthetic methods to provide efficiently with completely new nucleoside skeletons for further drug development. We could provide efficiently configurationally altered nucleosides by exploiting the thiolene coupling reaction, and we also provide diverse heterocyclic structures starting from nucleoside dialdehyde. Some of the compounds have already been tested, and the others are waiting for biological tests. And our idea is to build, for example, the tricyclano or morpholine nucleoside hybrid compounds into oligonucleotide and probably these compounds can be found application in gene silencing therapy, but this is the future. And at the end, I thank the work of my colleagues and for funding, and also thank the good collaboration for people from Belgium, Czech Republic, and Hungary in the field of antiviral study, and also I'm very grateful for the collaboration in the field of anti-malaria study to Professor Brijesh Ratti and his group, and thank you very much for your kind attention.
Okay, thank you very much for this talk. So I think we have time for some questions. Maybe I start, since I'm the chair, a naive question. So you showed your nucleoside analog, which have activity against the SARS-CoV-2.
Yeah.
So they function as prodrugs. In other words, they need to be phosphorylated to be active, or do you know do they target the polymerase? And do you know this?
We do not know the mechanism of action of these compounds. Probably the target is polymerase, but we are not sure of it because we found that the cell protecting group, for example, enhances the activity. So probably the target is different, not polymerases, but some compounds having an ester function at primary position were also active. So these compounds probably serve as prodrugs and are phosphorylated. So further studies are needed.
So you eventually will try to grow resistance, or to collaborate with grow resistance against the compounds to map the target?
Yeah.
Okay.
Yeah.
Interesting.
Yes, definitely.
Very nice talk. So you have made large number of compounds.
Yeah.
So when you modify any group, is there any rationale, or I mean to say you get it tested and then you think, okay, let me modify this, or randomly it is done, or you give it to collaborator, they tell you this is the issue, so then you modify. So any rationale behind that?
At the first stage, we want to produce new compounds. I should say randomly, yes, because we were interested in new methods and the scope and limitation of the methods, and then after having biological results, yes, of course, we want to design specifically against the targets, and we'll modify the compounds, the selected compounds, properly to the targets. Yes.
Thank you. Excuse me. Hello. Yeah. First of all, very interesting chemistry. I have very fundamental question that how you characterize that your tricyclono compound, there's the NMR splitting pattern was good in the upfield region, and second, I think in the mass also you must have gotten the fragmented peak. So first of all, what was the yield, and was the characterization was easy process for you for the tricyclono compound?
Good question. The characterization of the tricyclic compound was not easy at all. Yes, of course, we used NMR spectroscopy first to determine the structure. It was good enough to determine the constitution of the structure, so we know that the expected tricycle was formed, but we have no information on the stereochemistry of the new chiral centers, and we were lucky to have a single crystal from one compound, and then we could determine the absolute stereochemistry of the new stereotriad formed in this reaction.
Okay, and have you thought of doing that chiral HPLC to get the enantiomeric excess? What is the purity, like it's 50-50 or you are getting some racemic?
It was not racemic. It cannot be racemic because we have two chiral centers in the starting compound. So it was good to avoid to have racemic compounds.
That's why I was asking, if you go towards the chirality to do the chiral HPLC, you will have a broader picture about the enantioselective version of your compound.
I can understand your point.
Yeah, I was telling, if you do a chiral HPLC of your tricyclono compound, you can get a enantiomeric excess with it, whatever is the, like if you are getting R in a major amount or S in a major amount of that compound.
Okay, but
stereoselective fashion I'm asking.
yeah, it was no necessary to use chiral chromatography because anyway we obtained a pure stereoisomers in all cases.
Okay. Yeah, thank you so much.
Thank you so much for an interesting talk. You mentioned some of these compounds have anti-SARS-CoV-2 activity. Will you define it further, whether it's survival of SARS or it's binding to the H2 receptor or its amplification or its immune function?
Only routine tests were performed with these compounds. We did not make efforts to till now to reveal the mechanism of action. The compounds prevent the replication of the viruses. The compounds could not kill completely the virus, but inhibits the replication quite efficiently. But we do not know how this inhibition proceeds.
Any other question? Yeah, one here. So maybe the.
Ma'am, in thiol-based chemistry part, generally use highly polar starting material, but in case you not obtain about more than 90%, so how can you isolate this compound, because this is starting material is highly also polar, so how can you isolate this compound, final product from it?
In which type of
Thiol chemistry?
In the thiol chemistry, the compounds were not so polar due to the added structure, which were alkyl thiols,
Yes.
and sugar thiols with in protected form. So the compounds were not so polar, and the purification was quite easy.
So you use generally column chromatography or other techniques?
We used flash column chromatography, yes. In the case of morpholine containing compounds, these were highly polar. The purification of the morpholine containing compounds was more difficult, but we could manage it, and we also used column chromatography.
Okay, so let's give a very big round of applause to Professor Bordas for a wonderful talk. Thank you. And so I have to announce a change in program. So we have extra talk. Dr. Inderjit Singh, PhD, is going to give a 15 minutes, 15-minute talk. So can I ask him to come to the stage, please? So I give a very brief welcome. Introduction. So, Professor Singh got his PhD from Iowa State University at Ames in Iowa in the USA. And then he had several appointments. And at present, he's at the Medical University of South Carolina in Charleston, South Carolina, also in the US, with quite several other appointments, including with NIH in DC. So I don't have the title, but you should have the title soon.
This one. Next one.
Yes.
This one. Yeah. Okay. Yeah. Good morning. I'd like to thank the organizing committee for giving the opportunity to present this talk. And I also like to thank my local host, Dr. Deep Kalra, who was very gracious, making our stay very comfortable over here. So I'm going to share with you that what we did in the last 2-3 years, and we described a compound, a gene, which is associated with COVID disease pathologies. And the major problem with the COVID disease was that multiple disease mechanisms, multiple organs are involved in it, and no way to understand which drug will function, which drug will not function. So that's the reason the lot hard work has not succeeded against the people who are infected with COVID don't have any drug. Okay, so this just to kind of introduction. COVID came to this world without giving us any knowledge. What's the epidemiology of this disease going to be? How it will be spread? What kind of disease it's going to give? And how going to impact the population? Okay, and people who are infected and get better, what are the outcomes they are facing. So as outlined over here, close to 1 billion people were infected, and how many millions people died, and then there out of the people who survived, 10 to 12% or 15% tend to have long COVID disease over months or years and that sort of thing. So there's a big challenge, a disease which comes with so much dysregulations. There is not an organ which is not saved. Almost every organ gets affected when the vasculature gets dysfunctional, becomes dysfunctional. So it's a multi-organ disease, multi-mechanistic disease, and it's a big challenge for drug development. Drug developments were designed to target one protein, one reaction. How you can target 100 or 50 or 200 different mechanisms? Okay, that's why we don't have that drug so far. So vaccine came around, gave us good help in terms of decreasing the infections, but people still get infected. Okay, some people don't take vaccine, or the virus is mutating too fast. Even at this time, this February 24, over 400,000 people were in new infections. Okay, so it's certainly a major challenge. So when we started this work, we said, okay, what sort of mechanism we should target? Okay, so we look at, as soon COVID infects the host, there's an immune dysregulation, and that does pneumonia and systemic hypoxia. So the whole body is deprived of oxygen. Then there is a inflammatory oxidative damage to the vessels. Then there are coagulation problems in terms of clot formation, which translate into heart and stroke. Then vascular inflammation, immune cell transport to the organs which are affected, and then also integrity of the organ is vessels are completely dysfunctional. With the result, you have the pneumonia for the lung issues and the brain edema, which you see. Okay. So we just looked at the overview. This is a cartoon in terms of showing this is the virus, SARS-CoV-2. This is specific receptor for it, and it binds to this with this little triangle called spike protein, and is internalized where it's amplified and for secretion into the system. Moreover, this virus spike protein also activates TLR receptors, and it gives this innate immune response. That's why we get the temperature early after infection and that sort of thing. So, first problem was how to study this virus. In the labs, you cannot bring the virus in. Only in the whole country, United States, there are 3 or 4 labs. They're mostly associated with the defense departments. You cannot study it. Okay. Next was, unless you have a cell culture or animal system, you cannot test the drug. Okay. So after a lot of thought process, we said since the binding of this virus depends on the SARS-CoV receptor, and this is let's look at the binding domain. So we look at the binding domain of this protein, and we got to synthesize this much part. okay, about this 81 amino acid called S1 peptide. So it's a dead peptide. So we can do those studies in the lab without any kind of concern about applications or infections in that spot. So next question was, will this peptide give the covert disease? Okay. So this was the kind of overall what we did. We solubilized this peptide in the saline solution, sprayed on the nose of the mice. Okay? And then followed it through what kind of disease symptoms this virus has. So first thing we have to ask whether it develops temperature, weight loss, immune dysfunction, gets lung issues, infiltration of cells into lungs, the lung vascular pathology, what happens to the vessels, then also looking at blood coagulation, thrombotic factors, also looking at the lung edema, pneumonia, and brain vascular disease and edema. So these are the major pathologies which give you all kind of symptoms in the covert disease. And so here I show you the temperature. By the third day of spray of this peptide, they start developing temperature, these mice. Also start losing weight. Okay? And by that time, we already had figured out what kind of compound could be effective against all those disease mechanisms. So you have to have one drug targeting 10, 20 different disease pathologies. So this is the compounds we were looking at, and we look at the efficacy of these compounds in the temperatures. As you see over here, the temperature comes down with either drug, and the weight they start getting weight, both in male and females. Females are more resistant than the males. Okay, this was the novel discovery. This was the novel discovery that one gene pathology can contribute to all the disease processes you see in COVID. Never expected it. Anybody you talk to, they will say, man, dream on, keep on dreaming. So what we found, that this gene is upregulated in this mice. And this gene regulates express-1 enzyme, we call it GSNO reductase, that regulate the nitrobolome in the body. Nitrobolome-mediated signaling pathway are becoming popular in the last 10, 15 years. Over than 100 proteins been identified. And we have studied in so many different models, whether hypoxia, whether it's immune dysfunctions or traumas, and in any case, these become dysfunctional. So overall, just summary, this is the gene, this protein is upregulated in covert disease. What this does, this decreases this metabolite. The function of this metabolite is vascular protection, anti-inflammation, antioxidant, platelet, anti-activities, and so many other activities. So as soon as this is upregulated, this compound becomes less in our body. All this leads to dysfunction of all these activities. Okay? So these are physiological activities. When this is not optimal, you see all kind of pathological activities on the other side. Okay, so just another summary over here. There's a nitric oxide is made by NOS, nitric oxide synthesis. Either it goes to this side, become GSNO. It's a carrier of nitric oxide in our body because this nitric oxide is not stable. This is the only stable compound in our body. It does all these biological activities by cysteine nitrosylation. If there's any dysfunction on this pathways, then it goes to this side, reacts with the superoxide, becomes peroxynitrite. It gives you all kind of pathology you expect in that tissue. So it's the same slide which I showed you previously with the temperature and weight loss. Okay, so next was, let's look at this cytokine storm. That's we heard in the literature. As the virus infects, they say you get a cytokine storm. That gives you lung dysfunctions and others. Okay, so here we looked at blood cytokines, and they are upregulated in this. Okay, they are upregulated both in male and females, less in females than males. And when we use these two compounds, the levels of these cytokines become very low. Then you go further, look at multiple cytokines in the lungs, and they are upregulated and they are downregulated by these two drugs. So this compound controls the cytokine storm that you had, which is the mechanism for your systemic hypoxia. So another thing is where there's disease in any organs, these immune cells migrate to their organs to do the pathologies. So it shows that neutrophils and macrophages, they migrate into the lungs, male and females, and that these two compounds decreases that migration of the immune cells from the blood into the organ of interest. In this case is the lungs. And then it's not only innate immune response, even adaptive immune response. You look at cytotoxic T cells, they are upregulated, and males and females both downregulated with the drugs. Even the T cells, CD4 cells, they are upregulated, okay? Then they're downregulated. If you look at this is IL-17, one of the adaptive immune response mediator that's upregulated and that's downregulated with the drug. So this drug attenuates the immune response in the lungs. So next we looked at, just to summarize this thing, all these things I outlined are controlled or downregulated in this mice. Next one is to look at lung pathology. Does it really improve the lung function, lung pathology? You looked at cytokines, so what? Okay. Here we're going to look at vascular hyperpermeability, edema formation, vascular inflammation markers, the thrombotic markers, and fibrin deposition. So these kind of activity, we're going to look into it. And here, just look at inflammatory markers, 2 inflammatory markers in the vessels. These are vessels in the lung sections.
Sorry, Professor Singh, may I very humbly, because we have, I think, other speakers who, and we already have 20 minutes.
I'll try to go quickly.
Thank you.
Okay, so this is just tells these inflammatory markers are overexpressed. As soon give drugs, they become vessels start looking like a normal. Okay, so this is the quantitation on the other side, and here look at thrombotic factors, blood clot formation. We looked at number of markers, the fibrinogen, thrombin, these complexes, and all this tells you that even it reduces the clot formation. Okay, and last is to look at lung edema and brain edema. Look at here. Controlled lungs look like this, and these lungs from these animals which are exposed to this peptide, we inject a blue dye. The blue dye just goes through the vessels into the tissue. They become that blue. That's the pneumonia you see in patients, and this is protected by this drug, and this is the edema quantitation drug. And this is fibrin deposition. You see a lot of that. That's very reduced by the drugs. And here we also see that edema in the brain. This is a lung which I already showed you. Even the brain from these mice starts showing the blue color. That those vessels are all very permeable. And this is the quantitation, lung versus brain edema. And so this, what I try to explain to you, all the disease pathology we see in this mice is mediated through one gene. Then ask the question, if we take the gene out, so there is no increase in gene, will be protected. And so here's the mice, we took the gene out. In control mice, when we exposed to this peptide, look at the pathology in this mice, and both neutrophil and macrophages infiltration. Look at this knockout mice. They don't become sick. The immune cells don't go there. And same story is in the blood vessels, vascular integrity. These are normal vessels. This is the vessels of normal mice after exposure to this peptide. In the control, in the knockout mice, the vessels are intact. Okay? So this is fibrin deposition. Again, the knockout, you don't see any fibrin deposition. So this is a summary that this one gene gives the phenotype of COVID disease. And the inhibitors of that provide protection. Its disease is caused by a single gene, so far what we know. And so far, this is the only compound known which can target so many mechanisms of the COVID disease. So, the same thing. So, my message is, it's so unexpected that one gene can cause such a wide set of disease pathology. And target one gene with one compound can provide protection.
Thank you.
Okay, many thanks for this talk. So I think I invite you to approach Professor Singh individually for questions because we run over time. So, so let's thank again the speakers of this morning, Professor Boas and Professor Singh, with another round of applause. And I think we reconvene here in 5 minutes or what? 10 minutes. So 10 minutes means the next session starts at 10:45 sharp.
Now, I would like to request Professor Yogendra sir to felicitate our key speaker, Professor Indrajit Singh. Now I would like to invite you all for a short tea break. After tea break, we all are gathered here for oral presentation, Hall A for OP012, OP17, Hall B for OP18 to OP34 or Hall C for OP35 to OP52. Before that, all the delegates are requested to provide a video bite in which they are requested to say few words about ICBTM, about their research and their profile. I will encourage students of various universities to pursue sincere research and take part for forthcoming conferences. Thank you. And code number will collect it from registration desk. Thank you. All presenters, please load their PPT to our IT team. OP1 to OP17 for this hall. We assign the evaluator for Hall A, Dr. Prakasha Kampiyaha, Dr. Chetna Ameta, Dr. P.K. Chaudhary. For Hall A, OP01 to OP17. For Hall B, evaluators are Dr. Komal Sharma, Dr. Shikha Agarwal, Dr. Priyamvada Singh, and for Hall C, Dr. Vartika Jain, Dr. Mangi Lal Chauhan, and Dr. K.C. Pandey. Please, sir, join their presentation. Please check your attendance. Rakshit Ameta, Neetu Shorgar, Ajay Bagherwal, Anshuman Chandra, Chandrakant, Saurabh Gupta, Dipinder Sharda, Nidhi Sharma, Dr. Prabha Arya. Now we start our next session of oral presentation. I would like to invite Professor Dinesh Agrawal for chair the session of this oral presentation and invite Mr. Ankit Paliwal to felicitate Professor Dinesh Agrawal. Ankit sir, please felicitate our evaluator, Dr. Prakasha Kemapia. I would like to invite Professor R.C. Ameta to felicitate Dr. Chetna Ameta. I would like to invite Mr. Ankit Paliwal to felicitate Dr. P.K. Chaudhary. Thank you. Now I hand over the mic to Dr. Dinesh Agarwal.
Thank you, ma'am. Good morning to everyone. Before going to start oral session, we have to short talk. For that, first of all, I would like to invite eminent Professor S.C. Ameta sir for a short general talk. Over to you, sir.
Hello.
Welcome, dear friends. The topic of my short talk is, of course, it is away from the medicinal chemistry, but I wish to draw the attention of the younger generation towards two things, the two burning problems of the world. I have played my inning for 54 years as a researcher. So now the game is in your hands. I will talk about reduction of carbon dioxide to some value-added synthetic fuels. Next slide. Now, you know, global warming is a long term of warming of atmosphere on Earth. Though this warming is going on for a long time, but its pace has been increased in the last century due to burning of fossil fuels, due to transportation, industrialization, deforestation, and increasing population, which has increased many folds. Next slide. Now these are the greenhouse gases responsible for global warming. Carbon dioxide to the extent of 79.4, methane to 11.5, nitrous oxide to 6.2, others all chlorofluorocarbons, etc., to 3%. The major culprit of global warming is the carbon dioxide because it is 79.4, and whatever you burn, whether you burn coal, kerosene, gas, petrol, diesel, anything, it will add to some molecules of carbon dioxide in the atmosphere. Next slide. Now, another problem in the global warming is energy crisis. We are falling short of the energy resources. Our present traditional resources are mostly petroleum-based fuels, like petrol, diesel, etc. Now these fuels generate carbon dioxide, so it contributes both. We are facing two problems simultaneously. The global warming on one hand, and on the second, the energy crisis. It has been predicted that the traditional fuels will be exhausted within a few decades. So we have to find out some alternate source of energy. Or secondly, we can have a cyclic process so that neither the carbon dioxide is increased in the atmosphere, nor we are short of resources of the fuels. Next, please. Now, level of carbon dioxide is in 2004, 2023, last year, it is estimated that it will reach 496 gigatons in 2060. And we are facing a natural balance. For example, I will say this natural balance between human beings, animal, and plants is being disturbed because of many reasons.
transportation, road constructions, deforestation, etc. So there is a Newton's law. Every action has got an equal and opposite reaction. So nature has started taking revenge in the form of global warming. Next slide. Now what one can do? What is the solution to the problem? Photosynthesis was there. The plants convert carbon dioxide into oxygen, giving fuel as a biomass. Again it is burned, again carbon dioxide, revert back to oxygen and glucose, and this cycle was balanced. There is a natural balance between plants, animals and human beings. But because of our society, the growing society, it has been disturbed, it has been disbalanced, imbalanced. So global warming and energy crisis can be solved to some extent by a solution that is called photocatalysis in our terms. Normally the term photocatalysis is being confused. It is light catalyzed reaction. No. IUPAC has decided it is not photocatalyzed reaction, but it is reaction in presence of semiconductor. Light reaction in the presence of semiconductor. Now we want to mimic the photosynthesis. We want to prepare laboratory. In one of my articles on artificial leaf, there was an article published in Medical Journal on artificial leaf. We can have artificial leaves in the laboratory which can do the same job as the plants are doing in the nature. So process is going on everywhere. We are in the field from the angle of photocatalysis, where we convert carbon dioxide into some useful fuels. So carbon dioxide is controlled because we are utilizing carbon dioxide. Through photocatalytic means, we convert these to some value-added fuels, like formic acid, formaldehyde, methanol, methane, sometimes ethanol. So we convert carbon dioxide to useful fuels. Now formaldehyde, one of the fuel, is just precursor of glucose. It's the first unit of glucose. You add on formaldehyde, you will get glucose. So plants are doing it. We are not able to do it at present. But we can convert carbon dioxide into formic acid, formaldehyde, methanol, and ultimately methane. Next slide, please. Now, I have diagrammatically presented my idea. We are working in the field using some semiconductors. The coal, petrol, diesel, wood, gas, kerosene, when you burn these, they will add some molecules of carbon dioxide and atmosphere. Now through photocatalysis, in the presence of light, this CO2 from atmosphere can be converted to formic acid, formaldehyde, methanol, methane, which if burned in the fuel cell, it will generate electricity, which can be utilized for mechanical purposes, light, sound, anything. The same molecule, organic molecule like formic acid or formaldehyde, generate electricity at the cost of carbon dioxide and regenerate carbon dioxide again. So whatever you burn at present is adding to CO2, one molecule of CO2 in atmosphere. But this fuel, synthetic fuel, formic acid, because it was prepared from carbon dioxide, will revert back as carbon dioxide in the atmosphere. So this synthetic fuels will give you electricity, will give you sound, will give you mechanical energy, many things, and will not add even a single molecule of carbon dioxide in the atmosphere. If you take two molecules are back. 15 molecules, 15 molecules are back. 500 molecules, 500 molecules are back. While, on the other hand, our synthetic fuel, traditional fuels, add CO2 in atmosphere, causing global warming, resulting into natural catastrophes like melting of glaciers, rise of sea level. Maybe somewhere it has been predicted some of the countries will be underwater in this century. They will be almost underwater. They are only a few meters height. They will be all underwater because sea level is rising. So what I want to conclude is we can go for photocatalytic reduction of carbon dioxide in presence of a semiconductor Some of my students have presented the experimental data outside in the poster session. So I invite you all, particularly chemistry people, to find some solution of converting carbon dioxide into useful fuels like these. Thank you very much for questions.
Thank you very much, sir, for sharing deep knowledge with and regulating global warming and their effects and energy crisis. Now, I would like to call Professor Rameswar Ameta, sir, for their short talk.
Thank you very much. Oh, yeah, this one. Yeah. Thank you very much. Again, how the photocatalytic processes are there to solve the problem of energy crisis and environmental challenges. This is the very short talk on the use of nanostructure modified graphitic carbon nitride as an efficient photocatalyst. Professor Ameta has talked about the photocatalysis process, but which photocatalysis is gives the wonders for the processes of the reduction of carbon dioxide or splitting the water molecule into hydrogen and oxygen. Next slide, please. So this is the graphitic carbon nitride material. This is a wonder material just after the graphene, and this is actually having this two-dimensional structure, this tris as triazine, which is called heptazine also. So this is a wonder material which can perform various applications in the solving the problem of energy crisis and environmental challenges. Next slide, please. These are the characteristics of this graphitic carbon nitride. This is ultra hard material, excellent thermal and chemical stability, because various photocatalysts has been designed and used since last 30 years. But the stability is the main problem, and the stability problem can be solved by the help of using this graphitic carbon nitride based materials in the photocatalytic processes. Good heterogeneous catalyst, multifunctional metal free, because most of the catalysts we have been using since long are containing metal which has toxic effects. So all these effects are nullified by the help of using this graphitic carbon nitride wonder material, because it is metal free catalyst for clean energy conversion and effective photocatalyst. Adjustable, one of the important aspect of this is the band gap is 2.7 electron volt, and by functionalizing and changing the morphologies and so many other introducing the groups, we can tune the band gap. So the optical band gap is tunable, and 2.7 electron volt is a very good photocatalyst, because this it acts as a semiconductor, and there is a valence band and conduction band which are known as HOMO and LUMO, highest occupied molecular orbital and lowest unoccupied molecular. Now this 2.7 electron volt falls in the visible region. So the visible region or solar radiations can be harnessed by the help of utilizing this graphitic carbon nitride material. Next, please. But there are shortcomings, because 100%, nothing is 100% in the world. So their shortcomings are there. There is a low separation of excited carriers, photo-generated electrons and holes, because when the photons are allowed to expose on the graphitic material or any semiconductor, for example, titania is a very popular semiconductor used in the photocatalyst, zinc oxide and titanium, because of their stability. But their band gap is very high, 3.2 electron volt, which falls in the ultraviolet region. So the visible irradiation photocatalyst is used. We can use this wonder material graphitic carbon nitride, but the shortcomings are there because the separation of the excited carrier holes and electrons is very low. and insufficient light absorption, because in the visible region also the absorption is inadequate, because after 460 nanometer, 460 nanometer, it does not absorb. Small specific surface area in the pristine graphitic carbon nitride, and fast this is one of the most challenging problem, fast recombination of the charge carriers. Electrons and holes are recombining very fast, that's why the decreasing the quantum efficiency of the semiconductor. Next slide please. This is the mechanism how we can overcome these shortcomings by the use of coupling the semiconductor, this graphitic carbon nitride, with other semiconductors, such that whatever the hole is produced here and electron is reaching by the help of the light, this electron can be transferred to the conduction band of the other semiconductors, and the nanostructure of various nanostructures of these two coupled semiconductors, graphitic carbon nitride is also a semiconductor, and this other semiconductor is also there. So by the judicious choice of taking these two coupling of these two semiconductors, the various nanostructure graphitic carbon nitride based compounds can be formed, and their photocatalytic performance can be determined. Next slide please. These are various applications of graphitic carbon nitride material. This is a wonder material after graphene, or graphene is also a very wonderful material, but graphene band gap is 0. It is just acting as like a metal. So to convert the graphene into a semiconductor is another issue. We have to carry out some other processes, but luckily this compound is having another benefit, that it is already a semiconductor. This is used in organic synthesis, because the hole and it is a semiconductor, so the hole and electrons are there in the valence band and conduction band respectively, and organic synthesis for the redox chemistry, and even the coupling reactions. Suzuki coupling reactions can be carried out by the help of graphitic carbon nitride based nanostructure compounds. So this is a very important fuel cells. Here just kind of photocatalysis. What is the main important? Is photocatalysis. Various photocatalysis has been synthesized in the last so many years, but they have one or two, many shortcomings which can be nullified by the stability is the main challenging problem, because many times the chemical, due to chemical corrosion and photochemical dissociation of the catalyst, so the recycling processes are less recycling process. So stability is increased by the help of generating the synthesizing the nanostructure compounds of this GC graphitic carbon nitride. Photocurrent generation, the super photocurrent generation, because this has a very good photoelectronic ability. CO2 reduction, Professor Amata has recently talked about the CO2 reduction. Many scientists have tried in the laboratory, but they are unable to mimic the photosynthesis reaction. Millions of years are there, and these trees and plants are synthesizing their food by photosynthesis, but nobody is able to mimic this reaction in laboratory. Therefore, we can hope that for the carbon dioxide reduction into the value added fuels by the help of graphitic carbon nitride based nanostructure compounds can be done. Wastewater treatment, my two students are working on the dye degradation using these photocatalyst nanostructure compounds with another semiconductor, and we have obtained some good results in the case of Azure A and Azure B dyes. The hydrogen production is another very interesting and hot spot at present, because our Prime Minister has also launched a hydrogen mission, green hydrogen mission, and funds have been allotted. So, but the green hydrogen is not produced by this traditional way of getting hydrogen from the fossil fuels. So the green hydrogen is obtained by the splitting of water. Now splitting of water is another a challenging task, which can be further tackled by the help of using graphene carbon nitride, because all the shortcomings which we have highlighted, this is low, these recombinations of their low surface area, inadequate the absorption of the visible light, all these can be very easily tackled by the help of the nanostructure composites, nanocomposites of this compound. Various reports are there. one of my students is also performing this nanostructure by these binary and ternary structures, nanostructure compounds of the graphene carbon nitride. This inspection is very fast. In the USA and other Japan and other countries, the tiles are made up of TiO2, titania. Because titania, there are holes and the redox chemistry is working and the gram negative and gram positive bacteria is destroyed. But that is working in the field of UV light. So this can work in the visible light. So the tiles of the titania will be replaced by the graphitic carbon nitric compounds. Next slide, please. What the young generation, what my talk is designed for the young generation or chemist can do, because the rapid growth of the material science. If we look at the material science के अंदर सबसे ज़्यादा the very biggest the progress is there. And what a chemist can do, chemist can do a synthesizer or advanced photocatalyst which can mimic the natural processes in our laboratory, and for which we have to require well understanding of mechanism of the transport of the mechanism, because transport is very difficult. If the binary compound is there, binary nanostructure based compound of graphitic carbon nitride is there, there is a transport of an electron is very easy to understand. But if the ternary and quaternary nanostructure compound based graphitic carbon nitride based compounds are there, then it is in-depth understanding is required, and for that purpose what we have to do in situ technological process should be there to monitor that where the electron is going from one place to another. That in situ technologies require for the understanding of the transport of the by the efficient generation, photo generation of the electrons, their efficient separation and their accelerated transport mechanism of the electron will check the recombination problem, because by forming the nanostructure based compounds we can check all these shortcomings of this graphitic wonder material, graphitic nitride carbon nitride material. The main task is to develop suitable photocatalytic systems with high efficiency at low cost to realize a viable photocatalyst for different energy and environmental applications. Therefore, the young generation and my budding scientists are here. Now they can think over it, they can go through the literature and find which nanocomposites will be suitable, which advanced photocatalyst will be there, which will be low cost, with high quantum efficiency, and will be able to combat the challenging problem of climate change and energy crisis. Thank you very much for paying attention.
Thank you very much, sir, for sharing your deep knowledge with full of energy. Now, judges noted, I would now start with the oral presentation. And first of all, I request to code OP3, Dr. Rakshit Ameta sir, to present his paper on psychochemistry, the role of good and bad hormones.
Pointer नीचे, बस आगे पीछे, next or previous. A very good morning to all of you. Friends, in this presentation, I have not included any experiments or observations. Only I have prepared this presentation, short presentation, for your awareness, because we are talking about some medicines and drugs here. And don't you think if you find some alternative therapy which can help you to resolve your issues without taking any medicine? So let's see. Psychochemistry, the term itself reflects, means we are having many biochemical reactions in our body, and just by changing your thoughts, these biochemical reactions are controlled. If you are getting some good results, so you are happy, good hormones are released, and if you are having some bad news, some failures and rejections, bad hormones will be released, and accordingly your body and organs will react. So I will give you a short introduction about NLP, that is neuro-linguistic programming, how it can help you to resolve many of your health issues. Apart from my chemistry contribution in this university as professor of chemistry, I am honorary founder director of Total Solutions Samadhan, where we are providing different services to resolve many of the issues of in the society. So this talk is about awareness that I mentioned earlier, and especially in India, what I found, most of the persons are aware about the external world, but not much aware about the inner world, which is more important. If you are going to buy a car or a house, you will invest many amount. But what about your thoughts? What about your learnings? You will not take care of this thing. As I told you that in chemistry, not a single material in the environment which does not contain chemistry. So chemistry is everywhere. So as a researcher, it is contribution for the society, how we can contribute. These are some of the alternate therapies which provide you solutions for your health issues apart from allopathy. So it is an uncommon topic, of course, psychochemistry, introduction of NLP, but it is very important. It may be an eye-opening lecture for you. Here I would like to differentiate between the mind, types of mind. We have three types of mind: conscious mind, subconscious mind, and unconscious mind. Whatever information you are getting from outside through your five senses, this is stored in your brain in memory cells. There are neurons. So most of the persons think that this conscious mind is playing a major role in our life, but you will be surprised to know it is playing only 10% role in your life. Most of the contribution is given by subconscious mind, that is 50 to 60%, and of course 30 to 40% contribution by your unconscious mind. So neuro-linguistic programming, this concept was given by Richard Bandler and John Gridner in 1975. They found that this role of subconscious mind is very important in resolving many issues. Three terms are there: neuro, linguistic, and programming. Neuro means neurons, your brain cells. Linguistic, whatever information is stored there, accordingly you will plan something, you will tell something, you will thought something. That is linguistic, and your behavior will be decided by that. That is programming. So many of your health issues and many of the other issues also can be resolved by using this NLP technique. So behavior, whatever good behavior or bad behavior, we think that our thoughts are giving us this information whether I have to perform good or bad behavior, but you are not aware about your subconscious mind, which is playing an important role behind the screen. Here I have just given example of maturity. Sir, whenever my time is completed, just ring the bell. Yeah, thank you. So there are there are the three filters. We can say imaginary filters: deletion, destruction, and generalization. Whatever information you get from outside, first is deletion. Most of the incidents from your childhood we do not remember. So only important incidents are there in memory. Second is destruction. If we destruct some information and store in memory, this is destruction. And the third is generalization. Whenever you hear that a person is getting sick due to hypertension or sugar, diabetes, the medicine is required all over his life. So this is a general statement. If I ask you, who is the king of forest? If I ask you, who is the king of forest? Lion. Do you think that lion is aware that he is king? No. Who has given this statement? We. So most of the statement given in the society that are your belief system, which makes your memory. So I have given example of maturity. Do you know what is maturity? Most of the persons say that the person who is in more age or at a higher position may be a mature person. But in my opinion, a person who see all the aspects and then conclude a decision, he is much more mature person. Just I will give you an example. If you are having a friend and someone say that your friend is criticizing you there, so whenever someone will say someone is appreciating you, you may doubt. But whenever another person will say he is criticizing you, you will take immediately this statement correct. So whenever a person, another person tells you that your friend is criticizing you, you will have an image of your friend in your mind as a villain, isn't it? And your behavior will be decided by that image, not the actual image of your friend, which is not a bad man. So these are the, we can say, senses from which we store the memory. Our behavior, This is an interesting example. Normally what we see, tears are water droplets. आंखों से बहता आंसू क्या है? बहता पानी है. In a chemical sense, you can say it is having water, proteins, minerals, hormones, antibodies, and enzymes. Of course, with different chemical composition. But you will be surprising to see some of the photographs, how your emotions changes the chemical compositions. So, I have taken from online different photographs of tears in different situations. Tears of laughing. This is the same or same image of this dry tear.
Please, sir, conclude.
Yeah, tears of change, tears of grief, onion tears while cutting onion, basil tears, tears of possibility and hope, tears of remembrance. So you saw that at in different situations, the structure was different for tears. So emotions regulates your organs, and nowadays psychosomatic diseases are there. So doctors are unable to treat your organs because the problem is generated from your thoughts. Of course, these are the good hormones. You can remember those: dopamine, oxytocin, serotonin, endorphin. This is the flight and fear mode. Okay. Cortisol is the villain. This is a bad hormone. Many of the issues can be resolved by NLP that I mentioned earlier. So this was my just a small effort towards the audience to create awareness. Of course, as you remember 108, whenever you find some urgency for ambulance, calling ambulance, you can visit our website, and through Google you can search us. And what I mentioned, especially wherever you get help, you just go and take help, because timely help is very much important. So thank you very much, friends, for having your patience. Thank you. Thank you to the organizers.
Thank you, sir. Now session is open for questions. 1 or 2 questions only.
I wish I knew how to do it.
If there is no question, it is only 2 meaning. Either you have understood everything or you have not understood anything. But I am an optimistic person, so I feel that the 100% is grasped by you. So this is my contribution.
Thank you, sir.
Thank you. Thank you very much.
Now I invite next participant, Neetu Sorgar. Next is Parastak. Judges noted. Code is OP05 and topic is fluoride ions removed from groundwater, green approach.
Hello. Good morning, all of you. Today I present, my topic is fluoride ion removal from groundwater. It is a green approach. We know that, next. We know that water pollution is a serious problem at the present time. Light कौन सी? ये वाली. Slide change हो रही है. Light नहीं आ रही है इसमें. कौन सा वाला है? Light. अच्छा. Light नहीं आ रही है. Light इसमें light नहीं है. Pointer. Water pollution is a serious problem in the present time. We know that water pollution is due to many contaminants, mainly heavy metals, chemical, drugs, dye, pesticide. In addition, is also contaminated with fluoride in groundwater. We know that fluoride is a ion, and it present in rocks and underground water. We know that according to WHO and ICMR, Indian Council of Medical Research, if fluoride content is more than 1.5 ppm, causes fluorosis. Fluorosis is a serious disease. Many countries of the world are suffering from the problem. A variety of methods are used for fluoride removal, like ion electron method, osmosis, precipitation, bioabsorbent, etc. Out of these, the bioabsorbent method is inexpensive and simple, which can be used even in rural area. Fluoride considered in different states like Assam to Maharashtra. We focus on Rajasthan. In southern Rajasthan, fluoride problem in rural area is up to 5 to 6 ppm. We know that fluoride is reduced from the chemical element. We know very well can occurs naturally in water supply from amounts of dangerously high concentration. Fluoride is found naturally in soil, water and foods. It is also produced synthetically for use in drinking water, toothpaste, mouthwashes and various chemical products. Fluorine is a common element that does not occur in the element state in nature because it is highly reactive. Its account for about 0.3 gram per kg of the earth crust and exists in the form of fluorides in a number of minerals, of which fluorspar, cryolite, fluorspyrite. For the moment, the oxidation state of fluoride is -1. This is a fluoride concentration in agriculture product, wheat, cereals, rice, and this is the amount. A concentration of fluoride and biological effect. If fluoride in drinking water is 0.002 milligram per liter in air, injuries to vegetation 1 milligram per liter in water, dental caries 2 milligram per liter and more in water is molten enamel, 3.1 to 6 milligram in water is osteoporosis. If 8 milligram per liter in water is causes 10% osteoporosis, 20 to 80 milligram per day or more in water, creeping skeletal fluorosis, 50 milligram per liter in food or water, thyroid changes, 100 milligram per liter food or water, growth retardations, and more than 125 milligram per liter in food or water, kidney changes. If the acute dose is 2.5 to 5 gram, it means death. We know that the uses of fluoride in different products like rat poison, wood preservatives, mouthwashes, pesticides, glues, computer screen, tap water, used in good like toothpaste and mouthwashes, added to local drinking water, prevent caries, and mineral found in rocks, soil, water and air. Testing of fluoride by different method, fluoride ion meter, SPAN technique, unit is part per million or milligram per liter. Facts about fluoride, we already discussed the range is 0.5 to 1.5 milligram per liter. According to WHO, it is responsible for various types. Fluorosis disease. The question is, which is your water? Fluoride free water or fluoridated water? If fluoridated water is gives thyroid problem, lower IQ, fluorosis. Fluoride create many health problems like arthritis, osteoporosis, brittle bones, cancer, infertility, brain damage, Alzheimer's disease. This is the dental fluorosis picture. It is diagnosis by clinical examination, and it is a disease which mostly affected children 8 years old or younger during tooth development. The bone fluorosis, it is caused by mainly cryolite, Na3LF6. Some WHO report, according to WHO, World Health Organization, consumption of fluoride above the WHO concentration guidelines has more drastic effect, such as a tooth and a skeletal fluoride, which causes brittle bones. In 2012, Harvard meta-analysis 27 different studies found the children who lives in areas the high fluoridated water test significant lower IQ scores. We use the bioabsorbent. In the many literature, defluorination was studies using different bios such as a cyanoductylene based activated carbon, neem, kikar, charcoal, activated charcoal, bone meal, tamarins. We also use some plant product like Lantana camera, curry leaves, Ficus racemasa, Forania alumina, bell patra, removal of fluoride by different technique like ion selective method, carbon blockage criteria, thin film composite, reverse osmosis precipitation, adsorption ion action bioabsorbent. We use in this presentation we use Bili Patra. Bili Patra was used a low cost biomass used for bioabsorbent for defloration groundwater. So it is the effective for 5 ppm solution. We know that Bili Patra is used in Lord Shiva worship. The experimental part, material method, result discussion, isothermal study, kinetic study, physical characterization.
Please, sir, conclude.
Yeah. This is the effect of pH. We know that at pH 7, the efficiency is very good for removal of the fluoride. So it is used in neutral medium. Effect of absorbed dose, effect of contact time, effect of temperature, effect of mass size. It is very good for 30 BSS mass size, 62% removal. The isothermal study, it follow the Langumer isotherm because R square value is 0.993. The kinetic study, this adsorption follow the second order pseudo secondary reaction. Physical characterization, the same image. This is the before treatment, and after treatment this fluoride ion additive on the surface of bioabsorbent. EDX value, ion chromatography, here is no fluoride concentration, and after treatment this is fluoride concentration 6 point something. The conclusion is this: the commercially available absorbent can be replaced by the cheap and effective low cost absorbent. This is the reference, and thanks to Professor Suresh Ameta, Professor BP Sharma, Saras Kothari sir, Professor KK Dave, Hemant Kothari sir, and my scholar Dr. Akshay and Dr. Sundarlal. Thank you so much.
Thank you, sir. Any questions? Now, next participant is Abhisang Chandra. JJ is noted. Code is OP08. And topic is oxidative stress induced albumin modification, a prognostic block marker in the patient with fatty liver disease.
Very good morning to all, and respected chairperson and all the participants and faculties member. So I'm very privileged that I got the opportunity to present my research work with the IC BTM 2024, and my topic of research paper is like oxidative stress induced albumin modification, a prognostic biomarker in the patient with non-alcoholic fatty liver disease. So next. So just to cover, in a brief that what is a non-alcoholic fatty liver disease, you can consider that it's the most chronic liver disease globally, and the major factor is associated with the metabolic syndrome, obesity, insulin resistance, and the hyperlipidemia. Most of the people have shown that once they have gone for the biochemical analysis like triglyceride and other things, you can consider these are the major factors which is associated with the fatty liver disease progression and all. So the risk factor is the much higher in the person who has the diabetes, type 2 diabetes, and the obesity. And this is also a very alarming situation in the adolescent, the child. In the childhood, the children, they have the more obesity, they have the more chances to get a high fatty liver disease and the progression. till now, the only the method of the to diagnose the fatty liver disease is the liver biopsy, which is very painful and very difficult to understand the disease pathogenesis and the progression. So next please. So coming to the pathogenesis and the development of the disease, there are several hypotheses, and the proposal has been accepted by the clinician and the scientists. There is a multiple hypothesis hit and the two hit hypothesis. One, we can consider the stepwise disease progression. One hit is considered as the environmental factors and the genetic factors, which is plays a very important role, like family history of the diabetes, hypertension and obesity, which is the major factor of the first hit. And in the first hit, you can see the fat accumulation in the hepatocytes and the liver cells. The second hit start with the oxidative stress, because the other environmental factor which is influence the oxidative stress in our body. Now the question is that oxidative stress is a cause or is an effect, which is the major important part, which is playing the major role in the non-alcoholic steatohepatitis. Next please. So as you know that in our body the major plasma protein is the albumin, which has the several function to maintain our body system. But the quality and the structure, if you will consider, is a major plasma protein which has the highest molecular weight of like 66 kilodalton, and the tertiary structure, if you will see, is a very comprised and very systematic structure it has in our body. And it has the 35 residues, 16 residues. Only 1634 residues is free to maintain our osmotic system in our body, which is a very important part. If there is certain things happened in this modification, the activity of the antioxidant, the other things is hampered, altered in the condition. And if we will talk about the albumin function, the other like immune modulation is the major part, and the osmotic pressure, that all are the blood vessels, and the flow of the blood is important by the albumin things. And the major part that we can consider is the antioxidant, that we are talking about the oxidative stress and the disease progression. Next please. So based on this background, so what we really wanted to see, what are the albumin structure and the function of this form in the patient which has the fatty liver disease. So based on this, we have planned the experiment that we have recruited the fatty liver disease with the stage wise, like simple fatty stage patient and the severe non-alcoholic steatohepatitis patient, and the matched healthy control which have shown no liver history, liver disease background. So what we did, we have purified the samples, the albumin purified, and we have run on the ESI, electron spray ionization mass spectrometry, to see the isoforms of the albumin, and the cycler dichromatism is for the structural of the albumin. And we have seen the antioxidant activity to see the functionality of the albumin by the flow cytometry using the fluorescence dye on that. And we have seen the comparative oxidative modification in the disease condition as well as in the healthy control. Next please. So here is coming to my results, that we have recruited the patient and the healthy controls. If you can see the biochemical and clinical parameters, you can significantly find that the blood pressure, hypertension, diabetes, insulin, HOMA-IR, and ALT-ST, which is the liver marker in the normal routine testing, and cholesterol and triglyceride, you can find these are the biochemical parameters significantly high in the patient, which is showing the progression of the disease. Next please. So coming to the experimental plan, what we did, we have purified the affinity column chromatography to purify the albumin from the patient as well as the healthy controls, to see the structural changes and the functionality of these purified albumin. Here you can see this is circular dichromatism is showing that the changes in the secondary structure and the tertiary structure of the albumin. Here you can see that there is a tertiary structure or secondary structure has been changed in the albumin, which has the purified by the patient. Next, please. And this is the ESI spectrum by electrospray ionization coupled with the liquid chromatography. Here you can see the spectrum of the albumin. If this is the broad spectrum, you can see once it's run on the positive ion channel, it can be ionized in the protein, which is the specific ions. And once we have this calculated, we can convolute it in terms of the molecular Dalton. That here you can see the 4 isoforms. I mean 1, A, B, C, D. Here the peak intensity and the molecular mass is representing that there is a certain addition of the molecules in the albumin isoforms or the deletion of the albumin in that. So here you can see clearly in the healthy control, A is representing the healthy, B is representing the patient samples. Here you can see A, B, C, D, and here in the patient A, B, C, D, you can see the intensity of the peak, which is showing that these isoforms is higher in the healthy control, and this B is the higher in the patient. So what was that? These are the isoforms which is showing this 66.4 kilodalton is representing the cysteine residue is free. And if there is some addition, that means there is a binding of some molecules which is hindering the activity or ROS activity or oxidative stress activity in the patient. So here you can see clearly that the native albumin, we can say the functional albumin is the higher in the healthy control, but the cysteinated, which is the oxidized or disulfide bond formation occur in the patient, which is showing the higher oxidative stress marker in the patient.
Please conclude.
Next please. So this is after that we have just seen the antioxidant activity. So what we have done, we have taken the purified albumin, we created an in vitro model system to give a S2O2 oxidative stress, and we see the oxidative antioxidant activity. Then we have, you can see here 51.5 and 60.3, which is showing that the patient albumin is not able to decrease the ROS production in the next please.
It's my humble request to conclude.
So conclude this that the ESI mass spectrum has shown the 4 isoforms. 3 is the major forms, native albumin and the cysteinated albumin and the glycated albumin, and the other is the in vitro oxidative stress showing the reduced antioxidant activity in the patient. So to conclude my summary, that the measurement of cysteinated albumin in the suspected NASH patient might prove to a useful marker to assess the oxidative stress condition and the inflammation and severity of the disease respond to the treatment. Next please. And my request is to please take care of your liver, because this is a very important organ in our body and it has many role in the body to maintain. Thank you so much.
Thank you, sir. Any questions?
Thank you, sir.
Thank you, sir. Now next participant is Ajay Bagerwal. Is there? Next is Ansuman Chandra. Judges, please note it. Code is OP10. Ansuman Chandra, and topic is identification of novel compounds from Europhia, molecular docking and molecular dynamics study.
Hello. Morning, everyone. My topic of presentation is identification of noble compounds from olive. Oleoriopa is the common name is olive, जो हम खाते हैं जो normal. उसका database है, which is OliveNet. So what we did is we took the database of olive compounds and screened against an essential drug target of cancer, which is H-2, human dihydrouridine synthase. So let's start.
Next slide, please.
Next slide. ये चल नहीं रहा.
Yes, next slide.
Okay. Cancer. So, as we all know, cancer is a disease where a single cell fails to follow the apoptosis pathway and spread uncontrollably. Current estimates is like by 2025, we'll be having 19.3 million new cases. And this is largely due to lifestyle shifts and increase in life expectancies. Also, the carcinogens like chemical, tobacco, alcohol, UV rays are the lead causes of cancer. So here is a graph where we can see the male incidence and the female incidence per 100,000 cases, where we can see during the year 1990 and 1995 there was a peak. So the new new cases registered in 2023 in US. Here we can see all the cases of male and female, and in our study we focused on leukemia, which is 4% in males and 3% in females. These are the estimated deaths. For the case of leukemia, it is 4 and 5%, 4% in male and 5% female. Leukemia is a cancer where the blood forming cells creates abnormal cells, and here we see differentiated abnormal white blood cells in the blood. So approximately every 3 minutes in US, blood cancer is diagnosed. As of 2020, one out of 1,000 people are living with leukemia. For the case of children and adolescents, it is observed that leukemia are usually reported in case of children and adolescents. Cases of leukemia for individuals above 20-25 is rare, but they are present. So what we did is the drug target, which is recognized, it is a recognized cancer drug target. It changes the tRNA structure. Now tRNA is a transfer RNA which are highly modified post-transcriptionally, and here we can see the modification at different positions of tRNA, which provides which helps the biological phenomena like alternative folding, gene expression, translation, prevents frame shifting, and proper reading. Proper reading of the messenger RNA is very crucial for a proper frame protein expression. The most abundant mutation, post-transcription modification, is conversion of U to D, which is dihydrouridine synthase. Dihydrouridine is present in the D loop of tRNA, and it enables us for many avoiding base stacking in tRNA. Here what the reaction means, what does is it converts uridine to 5,6 dihydrouridine, where in the A section, A part of the figure, I've shown the double bonds break and addition of 2 hydrogen take place. The uridine and dihydrouridine image where the conformation of uridine and dihydrouridine changes. This is the first reported tRNA of yeast. It was reported in 1975, which has shown the conversion of D, means where the conversion of D is taking place. DAS is an enzyme, multi-domain enzyme. For prokaryotes, we have 2 domains, Tim-barrel domain and the helical domain. In the Tim-barrel domain, we have a cofactor binding position for FMN in prokaryotes and NADPH in eukaryotes. If we compare prokaryotes and eukaryotes, there are 3 domains in eukaryotes, as in humans, Tim-barrel domain, helical domain, and double-stranded RNA binding domain. The RNA binding domain in eukaryotes is very specific for tRNAs, and it provides a huge amount of specificity to the protein for the conversion.
Please conclude.
So the methodology where we used a database of, okay, let's conclude. We screened the database in 3 steps. At the first, we had 3,000 compounds, which were filtered down to 5 compounds. Here are the scores and the structure of the compounds. We saw the interaction where the substrate was binding to the protein and our ligands were directly interacting to the protein at that site only. These are the structure. Finally, we proceeded the ligands for MD simulations to just assess the stability of the protein in presence of the ligands. And it was reported, it was observed that the proteins was very stable and these ligands could be taken for scaffold selection and further optimization of specific drug. These are the interactions. And finally, we had some energy. So for concluding, we had a compound which is very specific for the DAS protein and could be taken for in vitro validations. Thank you.
Thank you, sir. Any questions? Thank you, sir. Now, next participant is Chandrakanta. Is there? Next is Saurabh Gupta. Saurabh Gupta. Code is OP12 and topic is pattern of hepatic dysfunction in scrub typhus patient in Udaipur.
Very good morning to the Honorable Chair and the audience present. So, myself Dr. Saurabh Gupta, I am faculty in the Pacific Medical College, and I usually we are used to present to an audience full of clinicians, but I am equally honored to be present here in a hall full of scientists. And I'll be starting my presentation. I'll keep it brief and stick to the facts which are relevant to the audience present here. So we started this study of studying the scrub typhus patients at Pacific Hospital who were coming in last year in the season of scrub typhus from July to November, December. And we found a similar pattern in almost all the patients of having a high amount of liver dysfunction in these patients. So our study is the study of the biochemical and clinical parameters of the patients coming to our hospital with scrub typhus, and we'll be focusing on mainly the liver dysfunction part here. Not working. Yeah. So we started this study under the valuable guidance of our respected Dr. S.K. Verma Saab, eminent professor in our institute, and my colleague in biochemistry department, Dr. Mukul Sharma. Okay, thank you. Now, a brief about the scrub typhus. Scrub typhus is a disease. It is a zoonotic infectious disease. It means it is spread via intermediate vector. Though humans are an accidental host here, and it is usually seen in cattle and mammals. And so it is a disease presenting with mostly with fever, and human beings are accidentally infected. It mimics other prevalent diseases like dengue, malaria. So dengue, malaria, leptospirosis are yes similar illnesses as SRE, and they present with fever and other constitutional symptoms. So Udaipur mein the prevalence and incidence is very high because it is a hilly area. It is an agricultural and hilly area. So surprisingly, Udaipur is a hub for scrub typhus patients. Now we see usually on a daily basis patients come in with fever, myalgia, arthralgia, headache, and gastrointestinal symptoms are the common symptoms here. And hepatitis with or without jaundice is also a very common findings in these patients. So our study was a retrospective observational study, and we included patients who are positive with an ELISA IgM positive test who are admitted in the Pacific Hospital from July 23 to November 2023. And the data was collected from the inpatient medical legal records, and the biochemistry lab record were also taken. Ethical approval was taken in due time, and we included patients of more than 18 years of age and tested positive for IgM antibodies against Orencia or scrub typhus antigen or scrub typhus organism. Excluded the patient who are having other causes of fever like malaria, dengue, and pneumonia and UTI, and who are negative for IgM scrub. Now the primary data collected was the patient with IgM or IgG positivity of scrub typhus by ELISA were included, and scrub typhus serology was tested for IgM for diagnosing the disease. The demographic and detailed history was also collected along with the past treatment history from the patient records. Relevant examinations, vital signs were recorded, hematological and biochemical investigations including CBC, blood sugar levels, renal and liver function tests, and electrolyte levels were collected. And other illnesses were excluded by relevant investigations. Now, what are the results which we got in our study? So we can see the majority of the patients lied in the middle age group, that is 30 to 60 years of the age. Almost equal distribution in the male and female, as males slightly more, 51%, and mostly the patients were from rural background. Now, why the patients were from rural background? Because this disease is prevalent in the areas which are near to the hilly areas. Just like Udaipur, it is surrounded all around by the Aravalli Hills range, and the patients who are working in agricultural lands or who are farmers by occupation or who have a livestock in their house. Now we collected the clinical profiles of this patient. So it is obvious that the patient will be presenting with fever. Some of the patients were not having fever, and the causes were due to some coexisting immunosuppressant illness. And surprisingly, the patients were having majority of the patients were also having cough. So I don't know the exact reason of this, because I think post-COVID the etiopathogenesis or immunopathogenesis of most of the disease has changed. Now we see typhoid patients, urinary tract infection patients also coming with cough. The present syndrome is cough in that. And myalgia was also there, and this is the hallmark symptom. This is the red flag in this patient, because patient coming with abdominal pain were also having high incidence of deranged liver functions or jaundice. 29.4% were clinically having jaundice or icterus. Now coming to the biochemical profile of these patients, you can see AST or aspartate aminotransferase levels were elevated in almost 70.5% of the patients in our cohort, or out of 85 patients. Similarly, ALT was elevated in 67% and respectively ALP levels. So, these three are the major liver enzymes which are seen in a patient of jaundice or hepatitis. So, elevated in more than 50% of the patient and reaching up to 70%. Bilirubin were also raised above the normal reference range in around 47% of the patients. Now, seeing the previous literature, the work done in this field, we have some very excellent papers from our country. Some papers from Uttarakhand, they described hepatic dysfunction in almost 51% of the patient, which is the highest percentage of the organ system involvement in scrub typhus patients. Similar papers were seen. This is a very large scale study conducted in almost 4 to 5 states, 250 plus patients. They also reported that the complications and death were higher in the patient who had primarily raised bilirubin and aspartate transaminase. A paper from Rajasthan in which 66 patients were studied, and the most common, they took patients who were having fever, 100% of the patient, and similarly here you can see hepatic dysfunction were also present, almost 50% of the patients here. Now, most of the work outside India has been conducted in Southeast Asian countries as well as in China. So, I took this paper from Taiwan, which is almost a similar study. They took 30 patients, out of which 29 had liver dysfunction, amounts to almost 90% of the patients. So, what do we concur here? So, this study is in the initial phase of the analysis. The statistical analysis is like dengue, malaria and typhoid. So, we have to look for the red flags or the signs like jaundice, abdominal pain and icterus to isolate or to initially segregate these patients who are at high risk of complicated scrub typhus. So, deranged liver functions commonly seen, we have seen almost 87% patient having deranged liver functions, and IgM antibody against scrub typhus is effective and a quick diagnostic methods. Incidence of hepatitis and jaundice is quite high. It is followed by respiratory and renal complication, which we have not studied in detail in these patients. It amounts to a very high mortality due to the patient, this patient can land into permanent liver failure. At this point of stage, it is very difficult to for survival of a patient, to get back the patient. Now, prevalence is high in rural areas and agricultural households. Therapies, yes, your primary therapy should be directed towards hepatoprotective. Your drugs should be inclusive of all those things which are beneficial for liver, for hepatoprotective therapies. Okay, and correlation with other complications like pancytopenia and nephropathy needs to be determined, which is the second phase of our project, which we will be taking in some further. This year also we are conducting studies on scrub typhus patient, which will include correlation with other systemic complications. Now, concluding my talk here, hepatitis can be an early differentiator in patients of scrub typhus because fever is present in almost all other similar illnesses, fever, myalgia and cough. So, hepatitis patients coming with a long standing fever, think of scrub typhus. Timely monitoring of liver functions and treatment can reduce morbidity and mortality, and empirical treatment can be done at peripheral centers with specific antibiotics, क्योंकि peripheral centers PHCs पे ज़्यादा investigations available नहीं होती, लेकिन yes, LFT and renal functions are available at very small centers as well. So वहाँ पे patient अगर 7 दिन का fever लेके आ रहा है, आपके पास liver functions है, jaundice के साथ patient अगर आ रहा है, so at that point of time they can administer empirical treatment as well there without getting a positive scrub typhus IgM serology. Patient can be saved in time because timely the drug of choice is doxycycline. अगर आपने टाइम से डॉक्सीसाइक्लिन इनिशिएट कर दिया, इट इज द चीपेस्ट एंड द इजीएस्ट टू गिव एंटीबायोटिक अवेलेबल, लेकिन इट इज लाइफ सेविंग ड्रग्स फॉर सब टाइफस पेशेंट्स. So the patients with severe liver dysfunction and fever of unknown origin, we should take scrub typhus always into consideration. And my talk here, thank you so much for patient listening. And I also invite anyone from the Pahar University or MLSU who are interested in doing clinical research, they can contact me. We can do an integrated study in these patients of fever or scrub typhus. Thank you.
Thank you, sir.
Any questions, please? Yes.
It's a nice case and nice presentation. It looks like I'm new to this scrub typhus.
Okay.
Is it a parasite or what is it?
So previously it was included in the parasitic genome. but now recently they have changed the classification and labeled it as a gram-negative bacilli or a bacteria. But it's transmitted by a vector mite, sugar mite. Chota sa.
The one that you
Yes,
showed.
I
is a
showed in
mite.
the
Okay.
first
That's the
slide.
vector, yeah.
Can you put the first slide?
So what's the intermediate host?
Intermediate are usually animals, mammals, can
Any
be
mammals.
rodents,
Okay.
cattle. एंड जो उदयपुर में जो यूजुअली जिनके घर में गाय भैंस या शीप ये सब होते हैं सर, तो वहाँ बहुत प्रेवलेंट है.
Oh yeah. Okay. So this pathogen is detected in the bloodstream. Is it?
No, we usually detect the antigen in the bloodstream, and the rapid test kits are available to detect the antigen and the antibody in the body. And we conducted the study by using IgM ELISA, IgM antibodies against Orantia tsutsugamushi, which is the pathogen.
So is it the site of multiplication or the organ affected? Is
Actually, the beauty of this disease, or I can say the horrific part, is it affects every possible organ.
Okay.
It can affect the liver, the kidney. There's no endemic site in the body where it is located, but it is sir मलेरिया की तरह है. मलेरिया फिर भी हम बोलते हैं कि लिवर में स्प्लीन में जाके वहाँ पे जाके colonize होता है. Yes, liver, spleen and RBCs. But scrub, it can affect any organ. We have even seen patients who are having bone marrow involvement or having CNS involvement.
Does it enter any cells?
Yes, anywhere in the body.
It's a multi-organ.
Yes, but the prevalence in hepatic dysfunction, kidneys and respiratory tract. ये तीन organ system mainly involved रहते हैं सर.
Thank you.
Thank you, sir.
Okay, thank you, sir.
Now next participant is Dipender Sarda. Judges noted, code is OP13 and topic is green synthesized insulin infused biometallic nano subclusters as multifunctional agents for ROS squelching, antibacterial resilience and accelerated diabetic wound healing. Dr. Sika Verma is here. Dr. Sika Verma. Nidhi Sharma. Nidhi Sharma. Dr. Prabha. Dr. Himani Dasora.
A very good afternoon to all of you. I am Dipinder Sharda and I am pursuing my PhD under the supervision of Dr. Diptiman Chaudhary from Thapar Institute of Engineering and Technology. Today I will be giving a talk on the topic, the role of nanoformulations in diabetic wound healing. Let's begin with the introduction to wounds. You all must have heard about the wounds. What exactly is a wound? It is the break or separation in continuity of skin due to any physical, chemical or biological means. A wound can be classified into different categories depending upon the cause of wound, depth of wound, contamination and risk factors involved, mode of injury, site of injury, and the time period for which the wound prevails. There are different phases involved in wound healing. Although wound is a very short terminology, but the entire process of wound healing is very complex and involves 4 different stages that begin with hemostasis, followed by inflammation, proliferation and remodeling. All these phases work together in a sequential manner to heal the wound by regulating the secretion of different cytokines and growth factors. This is a schematic representation of wound healing. In the hemostasis phase, there occurs the clot formation which prevents excessive loss of blood from the body. In hemostasis, there is vasoconstriction, which is the constriction of blood vessels, which prevents excessive loss of blood during the injury. The next stage is inflammation. In inflammation, the pro-inflammatory cytokines are secreted. Moreover, in this phase, the bacterial load which is present at the site of injury gets removed by phagocytosis. The third stage is proliferation, in which the anti-inflammatory cytokines are secreted. Moreover, angiogenesis and neovascularization takes place in this phase. And the last stage is remodeling, in which the epithelialization and the development of new blood vessels take place. There are several factors which affect the normal wound healing. The most important ones are the diabetes, the presence of bacteria, and the Ross species. All of us must have heard about the diabetes. In diabetic conditions, there is a delay in wound healing due to several reasons. The first one is the pro-inflammatory phase is elongated, which prevents the conversion of pro-inflammatory cytokines into the anti-inflammatory ones. Moreover, in diabetes, there is the slow blood flow to the site of injury, which prevents the supply of nutrients at the tissue site. Here you can see there is a delay in wound healing. This is a normal time period for the wound healing, and in diabetic conditions it is delayed by this much time. It is from here where the role of nanoformulations come into existence. We need different drug delivery systems for delivering a drug at the tissue site. There are various nanoparticles like inorganic solid lipid nanoparticles, the protein based nanoparticles, which can be used for delivering the drug at the target site. The protein based nanoformulations are gaining attention these days due to several properties. They have sustained drug release. Moreover, they are target specific. They are biocompatible, have low toxicity, and are very easily distributed across the body. After undergoing all this literature survey, we have developed insulin protected bimetallic nanoclusters, for which we have used 4 different components that are quercetin, silver, copper ions, and the insulin protein. We have synthesized bimetallic nanoparticles using the quercetin drug. We have used these formulations because of their antibacterial and ROS-scavenging activity. We have used insulin because it is not only a growth hormone, but it also promotes the secretion of different pro-inflammatory cytokines. Moreover, it also helps in the secretion of different growth factors. Here you can see our body has different receptors for insulin where our drug can easily bind. This is how the drug synthesized by us can be easily binded to the cells. And we have used this drug for wound healing, antibacterial and ROS-scavenging applications. These are the basic characterizations that we have performed. First one is the UV visible spectroscopy, which confirms the synthesis of nanoformulations. Second one is the FTIR studies, which are used for confirming the interactions between the protein and metal ions, followed by the Raman spectroscopy. In the figure D, you can see there is a very slight change in the structure of insulin, which confirms that the drug is very much stable after the formulation of nanoparticles. In the figure E and F, we can see there is a sustained delivery of drug at the tissue site. These are the HRTM and HRTM images of the synthesized nanoformulations, which confirms the presence of silver at the core, and the copper clusters are present at the periphery, surrounded by the layer of insulin protein. And in the figure C, there is the EDS, which confirm the presence of silver ion and the copper ions. Once we are done with the basic characterization studies, our next step is to evaluate their biocompatible nature, if the synthesized formulations are biocompatible or not, for which we have done the MTT assay in both normal and diabetic conditions. And here we can see that the synthesized formulations promote the cell viability. Following the MTT assay, we have done the scratch assay to confirm the role of formulations in the growth of the cells. Here you can see that in the diabetic conditions and in normal conditions there is a enhanced growth of cells, and the growth is much higher in case of normal conditions. Thereafter, we have performed the antibacterial and ROS-scavenging activity. We have used 2 different bacteria, both gram-positive and negative, and calculated the MIC values for these. And it shows that for the gram-negative bacteria, the MIC value is at around 16 mg per ml, whereas for the gram-positive, it is around 32 mg per ml. This is a ROS-scavenging activity. Here we can see that with increase in the concentration of nanoformulation, there is a decrease in the ROS-scavenging. With this, I want to conclude my presentation. Thank you. This is all about the role of nanoformulations.
Thank you. Any questions?
Yes, sir. हाँ जी sir. Yes, sir. I have shown the biocompatibility slide. This draft D is for the toxicity assay, and it confirmed that the formulations are not at all toxic. Moreover, they are promoting the cell viability.
Nice presentation. What was the model used for measuring the wound size? Is
Sir,
it cell-based or the wound?
Sir,
Next slide, yeah. Next slide, yeah.
Sir, we have grown the cells in 60 plates, which are circular plates, 60 and we have made the scratch using a pointed object, sharp object, which makes the scratch, and we measure this change in the wound width of this scratch assay using the PowerPoint. Yes, sir. Yes, sir. Human epidermal keratinocytes. Yes, sir.
Thank you.
Thank you.
This is the last. Now the session is over. Thank you all. Yeah, OP13. Yeah, over, sir. Over to you, ma'am. Preeti, ma'am.
Now I would like to invite Professor S.C. Ameta to provide valuable certificates to our evaluation team. I would like to invite Professor P.K. Chaudhary. Please, sir, get your certificate. Now I would like to invite Praksha Kampaya. Please, sir, get your certificate. Now I would like to invite Dr. Chetna Ameta. Please, ma'am, get your certificate. Now on, we were going to start a workshop on role of stress management in psychosomatic disorder. I would like to invite Professor P.K. Chaudhary to please felicitate Dr. Deepak K. Salvi, Professor and Head PMCH Udaipur. We will take a break of 5 to 10 minutes. Please be seated here. Thank you. Good afternoon to everyone present over here. Now we are gathered here for a workshop on role of stress management in psychosomatic disorder. I would like to invite Ankit Paliwal sir to please felicitate Dr. Deepak K. Salvi, Professor and Head PMCH Udaipur. Dr. Deepak K. Salvi holds the esteemed position of Professor and Head at Pacific Medical College and Hospital in Udaipur. With a wealth of experience and expertise, he has contributed significantly to academia, publishing numerous articles in reputable journals. His research has garnered high citations, reflecting the impact on his work. Dr. Salvi's skills and expertise encompass a wide range of areas within psychology, including psychological assessment, psychotherapeutic process, psychopathology, mental illness, psychological testing, personality assessment, psychoeducation, motivational interviewing, therapeutic alliance, and clinical assessment. Now, I would like to invite Dr. Deepak K. Salvi to please proceed this session.
So good afternoon, everyone. Good afternoon. Okay. First of all, I would like to express my sincere thanks and gratitude to the organizing committee of this conference for providing us an opportunity to talk about the role of stress management in psychosomatic disorder. Now I request Dr. Varade sir to give a presentation on the theoretical underpinnings of the stress and its role in psychosomatic disorder. Dr. Varade sir.
हेलो स्लाइड पहला गुड आफ्टरनून myself Dr. Mahendra Varade. I am working as a senior psychiatrist in the Department of Psychiatry, Pacific Medical College and Hospital, Udaipur. I introduce myself normally as I am a lifetime student of psychiatry. It is important than anything else because there is a lot of patients, there are a lot of patients, lot of experiences we come across in day-to-day life, and there is a lot much to learn from these day-to-day patients, their families, then our intersectoral cooperation from our different departments. So much to learn from different segments of medicine, and that teaches me a lot. Therefore, I do consider myself lucky and feel that I am a lifetime student of psychiatry. So, I always feel that there is always a space for improvement. So, if in today's presentation you find any lacunae, any shortcoming, I would like you to consider and give me a benefit of doubt and think about this guy always have a space for improvement, right? The topic of presentation from the team of psychiatry and psychology is the role of stress management in psychosomatic disorder. So, next slide please. My area of discussion is stress of effect on human body. Next slide please. So, areas to discuss, I would be discussing about health, its actual conceptual understanding of health, then what is stress, sources of stress, the pathophysiology of stress, stress hormones, stress and psychosomatic disorders, and some physical and psychological symptoms of stress. Next slide please. Health, as defined by the World Health Organization, it states that health is a state of complete physical, mental and social well-being and not merely the absence of disease and infirmity. Now, understanding health, we need to understand three different components, that is physical, mental and social. Unless and until we understand this, we won't be able to understand the pathophysiology of different physical illnesses, different psychosomatic disorders, and then it won't be, we won't be able to be in a better position to treat the psychosomatic disorders. Next slide. Now the question is, why do we need to understand the effect of stress on health? Understanding the cause of effect relationship is important. As I already said, that unless and until we understand the cause and effect relationship of the stress and physical health, it will be difficult for us to deal with the treatment protocol more effectively. Next slide please. Society and modernization. Coming to the actual part of how the stress is going to affect human life. So, every era in the history has been characterized by some debilitating disorders. There was plague, polio, smallpox, eliminated over a period of time. Now, in today's modern society, we have a characteristic disease which is major contributor to the disturbances of one's physical, emotional and social health. Next, the condition is known as stress and has been declared by the medical researchers at Cornell University Medical College to be the most debilitating medical and social problem in the world today. So, this came after a period of so much of research, and they identified so many effects of stress on different physical and psychological systems, and then they come up with the conclusion that stress is going to be very debilitating, the component contributor in the occurrence and in the causation of the physical disorders. Next slide, please. But if you go back to the Indian medicine, go back to the Ayurvedic medicine, around 23 years ago, so it's like around 2500 years ago, Acharya Charak, who is considered to be the father of Ayurvedic medicine, states that Vishado Rogovardhananam Sreshta, which is written in the 25th chapter of Charak Samhita, almost 300 BC. So that is first or second century BCE. So approximately 25,000 years before, 2,500 years before, sorry. So it states Vishado Rogovardhanam Sreshta. That means if you consider all the disease producing factors, Vishad, that means the stress or klesh or stress, something which is created inside your mind and body, would be the most which will come on the top of the disease producing factors. So this is the contribution of Indian medicine. Next slide, please. Coming to the present scenario, so certain modernization. So we are living in a rapid urbanization where stress is unavoidable. So previously, when we used to watch movies and dramas, and they used to say this, love is in the air, and now there is a lot of discussions and people say stress is in the air. So we need to develop ourselves to adapt to it. Darwin stated the survival of the fittest. So whoever is having the ability to adapt to stress, whoever is ability to adapt to the changing environment, that person will survive. So we need to learn over a period of time that we will have to develop adaptive strategies and adaptability to the changing environment and how to handle stress. That will help us having a healthy life. Next slide, please. So now coming to the actual what is stress. So stress is actually a wear and tear our body and mind experiences as we attempt to cope with our continually changing environment. So day to day life, nothing is constant. What is constant is change. So it keeps changing, and as a human being as well as animals, as every organism, you would find there is continuous change in their adaptability, genetic components, their ability to adapt with the different changing environments. Even plants in the deserts, you will find they are developed in such a way that there is a less of perspiration and then small leaves, and there is stomata which is responsible to store the fluid. So stress is a natural phenomena. It is not that bad. We need to understand what and how it going to affect. Next slide, please. So by definition, stress is what? Stress occurs when the pressure grows more and it is greater than the coping resources of human. So when the resources gets exhausted, you feel stressed up. Next slide, please. So the question is then stress a very dangerous word? The answer is no. Actually, stress is an ingredient, essential ingredient for normal functioning of your life. If there is no stress, you will have a laid back attitude and appearance, and you will not try too much for the achievements and your development. So stress is important, but this is positive stress, and that is called as eustress. So positive stress which help us to remain alert, motivated, to face the challenges and drives and solve the problems. So that is helpful in day-to-day life. So you should be able to differentiate between positive stress and negative stress. And then the distress. When the stress is too much for a longer time, acting for a very long time, then it results when there is excessive demand on your resources. So then it starts becoming chronic, and it becomes disastrous for your life. Next slide, please. WHO data, this is very significant. Recent data suggests that the number of people living in hypertension in the world that is doubled between 1990 and 2019. So the count raised from around 650 million to 1.3 billion, which is a huge component. And they say that stress being the major contributing factor in this particular development of people getting from non-hypertensive to hypertensive. And I didn't have much of the detail to include here, but at the literature suggests that there are around 50% of the world's population they are not aware about they are pre-hypertensive, and in a very short period of time they will be hypertensive. They will come in this category of hypertension or cardiovascular disease if they don't take adequate measures and precautions. So nearly half of the people with hypertension globally are currently unaware. And globally the number of adults with hypertension are predicted to increase by around 60% to a total of 1.6 billion population. So that is going to be a huge number of people getting into hypertensive category. Next. Coming to the Indian scenario. So there is a recent paper presentation by Dr. Naresh Trehan, who is a senior cardiothoracic surgeon and chairman of and managing director of Medanta Group of Hospital, Delhi. So the research paper says that 50% of population above the age group of 40 are suffering from some other cardiac pathology. That means every second person of the crowd, every second person who is above 40 is suffering from some other cardiac pathology. We might be aware, might not be aware. So better you get aware and get yourself investigated, checked, and if there is a pathology or if there is a pathology in a primary stage, we better take care of it so that it won't lead to any complication in life. So Dr. Naresh Trehan's statement came as stress around everyday living taking a toll on people and contributing to a significant spike in cardiac events. That research paper says. I think this is very significant and alarming. Next slide, please. Next slide. No, this the previous one. This statement and investigation and research they came into existence. They started thinking about all Indian scenario after the sudden death of singer KK live during the live concert. He suddenly collapsed and he was taken to Medanta hospital and where he was declared dead. Then another victim was Raju Srivastav, the stand-up comedian who failed during this exercise in gym, and then he was admitted for a few days and then collapsed and succumbed to death. Then recent developments, the television actor Mr. Siddharth Shukla, most of us know it, those things. So it started shocking the nation. So there are few few names to say here, but undercover there are so many deaths recently happened secondary to cardiac illnesses, and that exacerbated after especially COVID. As per Indian Heart Association, they said disease strikes Indian at the very earlier age, and previously the age was found to be more above 50 years. Now they say it starts somewhere between 30 to 60. You have to start checking yourself from the age of 30. So that is very alarming, and that is very significant development of the research, stating that from the age of 30 itself you should be careful if there is any genetic predisposition. So at least they say if you compare it to the Western countries, we are at risk of at least 10 years before the people of the Western countries. Next slide, please. Again, the statement came from renowned psychiatrist cardiologist C.M. Manjunath, stated that India will gain a notorious distinction of recording the highest number of cardiac deaths by 2030. This is again very much serious statement, and you should be careful about it. So they say out of the total deaths, virtually every fourth will occur due to cardiovascular diseases, and stress would be a very major component in that. Next slide, please. Why Indians should be very careful? That is important to know, because the research paper said by published by Naresh Trehan says that we Indian, or most of the Asian population, we are genetically prone to develop cardiac diseases because there is a genetic damage to the intimal layer. Due to the genetic damage to the intimal layer, whenever we are acted upon by severe stress, there is a massive change in the intimal layer, and we are prone to develop cardiac diseases. Next slide, please. That is how the overall balance shifts from health to disease when you are acted upon by uncontrolled and chronic stress. Next. So where does the stress comes from? So there are so many things, so many areas, so many components responsible to push you into the stress. There is interpersonal conflicts, then social interactions, major life events like divorce and death of the near and dear ones, and then interpersonal conflicts at a family level. Now you can see interaction with your children. So many things, broken finances, day-to-day life hassles. So that leads to stress. So those are the sources you have to identify and start working on it. Next slide, please. Social interactions. Most of us might get an experience of rudeness and bossiness and aggressiveness by others, bullying, and your personality when you are given some responsibility and you can't say no. So sometimes you need to understand and learn to say no, because if you learn to say no, then the stress will be minimized. Next slide, please. I'm trying to just go fast through with few slides because of the time constraints. So organizational rules, regulation, then red tape and deadlines. Nowadays you might see the people working with finance and people working with different organizations. They are given deadlines. इतने दिनों में लोन होने चाहिए, इतने दिनों में इतना टारगेट सेट होना चाहिए. IT professionals, corporates, so they are under so much of stress. So that is the source of stress. Next. Family stresses, the marital discords, interpersonal relationship problems. The psychology department people are, our psychology department comes across a lot of family and marital discords. So we deal with those things on day-to-day basis. So most of us know, but it is important for the other non-medical population to understand these are the components which needs to be addressed properly. Problems in interpersonal relationship, already said. Financial demands, expectation from yourself that you want to have leisure life and lot of luxury, so you will have to limit your expectation in order to handle your financial demand. And then medical expenses. Next slide, please. So the sources of stress, major life events, death of near and dear one, this rates the highest in stress producing component. Loss of job, again very important. Transfer to different place, and then you have to adapt to the new environment, new people over there. So it is again stressful. Unless until you get adapted to the new environment, you are under stress. Then transfer. Promotion. Sometimes people say promotion is good, but why it is stressful? So that depends upon your personality, whether you are taking it as challenge and opportunity to develop yourself, an opportunity to have another. So that can be, you can be happy. But then if you take promotion as a responsibility, added responsibility, more and more expectation from yourself of the management, then it can be stressful. So you will have to judge it and decide accordingly. Then marital status change, these are the another issues. Birth of a new one in the family. Someone would ask you why birth is stressful. So it is the very happy moment, but then many a times the parents, they are not ready. Nowadays पहले तो ऐसा होता था कि शादी हो जाती थी, बच्चे हो जाते थे. अभी तो ऐसा प्लानिंग होता है. They have to plan for the child's future, and then the insurance and finance and education, वो सारा प्लानिंग होता है. बाद में बच्चे का प्लान करते हैं. But in between, before the planning, if the child birth occurs, then it is stressful. Sometimes we come across such cases. So even that is also stress producing. The retirement, again a person who is highly workaholic, served for years together, and now they are in a stage of retirement. So they are not mentally prepared that I will be absolutely workless, I will be sitting at home, so how can I handle? So I would suggest those who are planning retirement, they should have their future plan, how and where they want to accommodate themselves, and how to utilize their expertise, and their healthy aging will help the planning will help in retirement, so that won't produce too much of stress. Next, natural calamities. Many of the researches they are done at the earthquake areas of earthquake, flood, tsunami. People are devastated. Their entire life is completely changed. So those things are there. Then war situations. Now we are seeing so much of immigration from the war areas to other areas. So other countries are getting stressed up. Their finances. So many things happening around produces stress. So it's not only the area where the war is happening, but surrounding countries and so many other things are getting affected. So stress is getting developed, getting built up everywhere. Next slide. Then coming to, you have to identify your personality. There are personality traits, different personality types, and different personality deal with day-to-day life in a different way. Their way of handling, that is called defense mechanism, are different. Just type of personality, the people are impatient, time urgency is there, aggressive. So the moment the things are not happening according to their requirement, their vision will, their way of thinking, then they get stressed up. That creates frustration. They have deep-seated insecurities to deal with, and I would suggest if some people are coming across such situation, they should help. They should take professional help. They should seek help. Then type B personality, these are non-aggressive, relaxed, flexible in opinion. So they have realistic targets, and these people are able to handle stress in a better way. That is type B personality. So they have the better coping mechanisms. Next, type C personality. These people are like, they have different states like anxiety states. They are anxious, might be genetically, might be during upbringing, they become more and more anxious because of the some issues in upbringing. So anxious avoidant personality traits, so they try to avoid situations. So stress, the moment they are faced by unexpected sudden change, they get anxious and the stress builds up. So some people are perfectionists, as it is shown that the person is cutting grass, but he wants everything in a very proper way, so that creates stress. Other slide shows that person should be taking rest in the hospital, but he is so workaholic that वहाँ पे भी कुछ उठापटक करने की सोच रहे हैं. So these people can't remain at rest any time. So they create and generate stress for themselves. Next slide, please. Stress response. Coming to the pathophysiology of the stress. Stress response, 1930. Dr. Hans and Dr. Walter Cannon, they were the people who identified and done research on it. So it has got, it's called a general adaptation syndrome, which has got 3 phases. One is alarm response, adaptation, and exhaustion. Next slide, please. So alarm response, what happens actually in alarm response? This is called as flight or fight response, because it prepares your body for immediate action. Either you run away from the situation or you fight back. So combat situation, you are faced by some sudden danger and your body needs lot of energy to deal with it. So what happens actually during this sudden acute stress, on evolutionary basis, adrenaline rush is there, your brain is working, the adrenaline is pushed into your blood circulation, sweating, your raised heart rate. So heart rate इसलिए बढ़ता है कि it's pushing the blood into the circulation, so that if you want to run away, you will have enough of oxygen supply to your rest of the body. So increase blood sugar levels. So if you are running, your muscle need lot of sugar, and now you are ready for action. So that is the first reaction when you are acted upon by the stress. So that is how it happens. The first response is alarm response. Next, what if you cannot channelize the anger? Now we are not living in a stone age. Had you been living in a stone age, you had got 2 options: either to fight back, because there are no too much of social constraint, you can fight back or you can run away. But we are living in a stage of modernization with lot of social issues and social constraints. So what happens, you cannot either fire back, you cannot handle the stress, you have only option to stand there and face it. So the moment it happens at different places, interactions. So this is the condition where stress builds up in your unconscious, and you carry that stress throughout your life. Next slide, please. So if stress is generated inside your body and you can't fight, you can flight, and then stress becomes chronic, that remains, the stage remains for a longer time. In chronic stress situations, what happens, person enters into stage of exhaustion. So there is emotional exhaustion, physical exhaustion, psychological exhaustion. So your resources start slowly getting exhausted, your body starts becoming weak, and the continuous release of cortisol, it starts putting breakdown in your muscle and your protein, and your body starts becoming weak, and then the illnesses, the different immune system responses starts coming up, and then there is a possibility that you collapse. Next slide, please. So endocrine system, it is mainly responsible for the stress response. So stress response is controlled by endocrine system. This is called hypothalamo pituitary adrenal axis, HPA axis. So demand on physical and psychological system. So it really is results in hormone secretion. The next slide I will show how hormones are secreted. Next slide, please. So it's the brain. It has got a gland known as anterior pituitary. So there is a secretion. When you are acted upon by severe stress, the anterior pituitary, that leads to ACTH, adrenocorticotropic hormone. So that hormone acts on adrenal cortex. This is adrenal gland inside your body. So it leads to secretion of chemicals. So it leads to secretion of cortisol and adrenaline. So these are the predominant hormones or chemicals responsible for further reaction, because once the cortisol or adrenaline is pushed inside the blood, then it will have its own effect on the body. So what adrenaline and cortisol can cause, I'll let you know in the next slide, please. Yeah, here it is like the cardiovascular system, most of the body systems, they are under control of autonomic nervous system, and it works in correlation, in coordination with the previous system, hypothalamo pituitary adrenal axis. So the hormones, they work and they are released and they are stimulating the autonomic nervous system. So that is how the things happen. So if there is an excessive hormone, that will increase the heart rate. Then if the heart rate is increased, the more blood is pushed into circulation, so that will lead to high blood pressure. More sugar is pushed into circulation, that will lead to high sugar level. So that is hyperglycemia initially, then leads to diabetes if it is not controlled. Next slide, please. Stress and psychosomatic disorder. Coming to the psychosomatic disorder. So it was initially discussed and it was initially researched by Franz Alexander, Chicago Institute, in 1950 to 60. They say that psychosomatic disorder is a condition in which psychological stresses, they adversely affect the physiological component. Psycho means psychological and somatic. Physiological is somatic. Okay, somatic or physiological functioning to the point of distress. So what happens? Psychological conditions or distress leading to release of chemicals damaging your body organs. It is a condition of dysfunction or structural damage in the bodily organs through inappropriate activation of the involuntary nervous system, as we have already discussed the system and how the hormones are released. Next slide, please. So psychotic and somatic symptom emerges as a physiological component of the emotional state. Okay. So in the state of rage, example, in a state of rage, you find the person is angry, his blood pressure is likely to be elevated and his pulse and respiratory system are increased. When anger passes, so this acute stress, the person will have this kind of symptom. So when the anger passes, the heightened physiological responses comes back to normal. Okay. Now what happens if it continues? So one spurt comes, body responses, anger settles down, person becomes normal again. So achieves the normal physiological processes. Next slide, please. So what will happen if the person has a persistent inhibited aggression? You are continuously under stress, you have got rage against someone is continuously pushing you and you are having continuous aggression, that is called as chronic rage. So same physiological response will continue, right? The emotional state remains unchanged and though unexpressed in the overt behavior. हो सकता है कि बंदा आपको दिखाए नहीं कि he is under stress and under rage or he is having that aggression against someone, but unconsciously it is happening, and because of that psychological component, his body is continuously in a state of heightened alertness. So that will that will lead to continuous high blood pressure, heart rate will increase, sugar will be high, and that will start damaging the bodily organs. Next. So therefore it is called a psychosomatic. Consistent psychological pressures and stress leading to consistent hormonal and chemical disturbance in body leading to continuous damage, and slowly starts damaging. One fine day person starts that kidney is damaged, liver is damaged, and they say कि why it happened, don't know. फिर पता चलता है कि if you study it retrospectively you will find that there is a continuous stress and cortisol levels are very high. So stress affects psychology in different possible ways. One is direct effect. या तो स्ट्रेस डायरेक्टली डैमेज करेगा आपके बॉडी को थ्रू केमिकल्स, या इंटरफेयरिंग विद एग्जिस्टिंग डिसीजेस है लाइक डायबिटीज, हाइपरटेंशन, स्ट्रेस से वो बढ़ेंगे और इंटरफेयरिंग विद द ट्रीटमेंट प्रोटोकॉल. नेक्स्ट स्लाइड प्लीज. That then again stress leads to oxidative. This is very important just to understand. There is a release of reactive oxygen species, and these are formed as a natural product of the cell activity, and then when it continues to grow inside the body, it starts damaging the body structure. This damages your body on the cellular level, mitochondria and cellular level. So from there the disease process starts. कई बार पेशेंट को पता ही नहीं चलता कि सब कुछ नॉर्मल था, मेरा ब्लड प्रेशर नॉर्मल था, सब कुछ नॉर्मल था, अचानक से लीवर में कहाँ से प्रॉब्लम आ गया, अचानक से कैंसर सेल्स कहाँ से डेवलप हो गए, अचानक से वन फाइन डे दे कम टू नो दैट दिस इज अ स्टेज 2 कैंसर. फ्रॉम वेयर इट हैपेंड? Apart from the genetic component. So stress leads to genetic transformation, mutation in the cellular level through ROS, leading to the devastating, life-threatening diseases. Next. वो बटन दबाते रहो, वो आते रहेगा. Next. This is how it shows that this is how the oxidative stress leads to damage to different. This is how the cell phagocytic activation of the cell, and then cancerous cells develop, and then disease process takes place. Next slide please. Next, just keep pushing. This is environmental ROS. Again I said damaging the cell membrane and the mitochondria, producing the disease and aging, cancers, degenerative disorders of the brain and body that leads to that happens because of this stress. Next. Here it shows that normal cell, how it gets transformed into the phagocytic cells. See the borders of the cellular margins, and how it starts getting converted into the phagocytic cells, leading to cancerous development. Next slide please. Leading of free radicals. Next slide. Considering again brain, it affects oxidative stress, leads to Parkinson's disease, again degeneration of the brain cells and chemical disturbance in the brain. Next slide please. I am going very fast because we have time constraint, as well as you can understand how the pathophysiology and how it leads to pathological changes. So stress and Alzheimer's disease. So ऊपर केस में दिखाया normal aging, and नीचे केस में दिखाया this is abnormal aging because of this stress. How the tau protein and damaging the brain cells, leading to degeneration of the brain and Alzheimer's. So normal aging around 65-70 के आगे होता है, and if you are talking about the stress induced diseases, then the dementia can start early. Next slide. Again a very important area of stress related disorder, digestive disorder. Patient who have gastrointestinal disorder also have high psychiatric comorbidity. The data suggests the prevalence of depression is excess of healthy controls was found around 39% in ulcerative colitis cases and control trials, and around 33% in IBS, irritable bowel syndrome. So this is the data given by WHO. Next. This is attributed to the combination between, this connection between the gastrointestinal system and the brain. This is called as gut-brain axis. So this is how it gets affected, the system. Effect of stress, I will just go fast with this symptom, and I think the next speaker, Dr. Mehta sir, will be elaborating more on physical symptom of stress. Next slide. So physical symptoms, you come across sleep disturbances, fatigue, dizziness, then digestive changes, loss of sexual drive, headache. So these are the different symptoms you will come across when the patient is acted upon by severe stress or going through the stress. Next slide, please. Psychological symptoms like palpitation, panic attacks, chest pain, restlessness, dryness of mouth, depression, so many. There's a huge literature. If we start talking and keep talking about it, we won't have time. So I think coming to almost concluding my part of the discussion. Next slide, please. Karl Marx once said that to be modern is to find ourselves in the world that promises us adventure, joy, growth, and transformation of ourselves. But at the same time, it threatens to destroy us, destroy everything we have and destroy everything we are. So, to conclude my part of the presentation, I would like to say, as psychiatrist and field of psychiatry, the worst is gone, but best is yet to come in psychiatry. We as a professional of this field, we as a professional of psychiatry and psychology, and along with different associated branches, would like to commit ourselves to bring the best to the humanity. Thank you. Thank you so much. Time kids.
Thank you so much, Dr. Mahendra. Dr. Mahendra completed post-graduation in psychiatry from Topiwala National Medical College, Mumbai in 2001. They also worked as an assistant professor in psychiatry at Bharati Vidyapeeth, Pune from 2009 to 2014. They also worked as senior consultant in psychiatry at GBH American Hospital and PIMS Udaipur from 2016 to 2018. They also worked as a consultant and psychiatrist at different de-addiction centers in Chandigarh, Punjab and Haryana from 2018 to 2022. They were also an official member of doctor's team and psychiatrist. They had privileged work as central observer of SAI, Sports Authority of India, for National Anti-Doping Agency and being member of World Anti-Doping Agency in 2008. They were also appointed with Indian hockey team at Malaysia in 2008 and 2019 for Suburban Asab Shah Hockey Companionship. Now I would like to invite Dr. Deepak K. Salvi.
Thank you, Varade sir, for providing us the beautiful knowledge and insight about the role of stress in the development and maintenance of psychosomatic disorder. In this session, I would give a live demonstration of mindfulness practice, which is one of the most widely used stress reduction technique in the management of psychosomatic disorder. Before I give instructions on mindfulness practice, I would like to give a very brief introduction to the term mindfulness. The same slide, the same thing. Mindfulness. Yes. So the term mindfulness refers to the psychological state of increased awareness and the practices which promote this kind of awareness. So, yeah. Pointer. Okay. So, mindfulness refers to the self-regulation of attention. So it is a flexible self-regulation of attention and awareness, which cultivates your orientation towards, one's internal and external experiences in the present moment, which is characterized by the attitude of curiosity, openness, acceptance, and gentleness. Next slide, please. So, in the mindfulness practice, there are three core components. The first component is intention. Intention creates the central motivational factor, and intention sets the stage for what is possible. So intention is very important while practicing the mindfulness. The second important factor is the component is attention, which involves your awareness, which involves your observation to the one's internal and external experiences. And the last one is the attitude. So attitude describes one's quality of your attention for this, whether your attitude is harsh, whether it's gentle, whether it's curious. So we have to bring this attitude in the mindfulness practice to facilitate the awareness process to one's experiences. So these are the inseparable components, and without this, mindfulness practice is not possible. So now I would like to give a live demonstration of mindfulness practice. Before I start giving instruction on mindfulness practice, I request you all to put your mobile phones on silent mode. Shall I start? Okay. So let's start the journey of enlightenment. Settle yourself in a comfortable sitting position on a chair. Take a comfortable position, please, everyone. Allow your back to adopt a dignified and comfortable posture. Place your feet flat on the floor with your legs uncrossed. Good. Now gently close your eyes. Everyone please, now gently close your eyes. And bring your awareness to the level of physical sensations by simply focusing your attention on the sensations of touch and pressure in your body where it makes contact with the floor and the chair you are sitting on. Spend a minute or so to explore these physical sensations. Now try to bring your awareness to the changing patterns of physical sensations in your lower abdomen as the breath moves in and out of your body. As the breath moves in and out of your body. Here you can also place your hand on your lower abdomen and become aware of the changing pattern of sensation where your hand makes contact with your lower abdomen. You can also place hand on your lower abdomen in order to keep track of all the physical sensation around the abdomen. Good. So this will facilitate the awareness process. Now try to notice the movement of your abdomen. Try to focus your awareness on the sensations of slight stretching as the abdominal wall rises with each in-breath and of gentle deflation as it falls with each out-breath. जैसे जैसे पेट फूल रहा है, पेट अंदर जा रहा है, उसके सेंसेशन को आप फील कीजिए हाथ से. सांस अंदर जा रही है, पेट फूल रहा है, सांस बाहर जा रही है, पेट अंदर जा रहा है. Try to feel the sensation of your abdomen. Here you can also follow with your awareness of breathing, awareness of the changing physical sensations in the lower abdomen all the way through as the breath enters your body and all the way through as the breath leaves your body. Feel the sensations, changing patterns of physical sensations. Continue paying attention to your breath as it enters and leaves your body. There is no need to try to control your breathing in any way. Simply let the body breathe itself. You can also bring this attitude of allowing or letting go to the rest of your experience. There is nothing to be fixed, no particular state to be achieved. Very good. Try to concentrate on your breath. Now, sooner or later, your mind will wander away from the focus on the breath in the lower abdomen to thoughts, planning, daydreams, drifting alone, or whatever. And this is perfectly okay. It is simply what minds do. It is not your mistake or a failure. Don't try to judge or label your effort to mindfulness practice. Just try to be non-judgmental. Do not try to label any of these sensations. Just observe them as they are. Don't try to push away and avoid the thoughts or images if you get caught up in this passing event. Because your thoughts and images are just passing events. Don't try to judge them. Don't try to label them whether they are good or bad. Just observe them. These are just thoughts. These are just mental passing events. Let them come and try to experience them as they are without being hijacked or caught up by them. Often our minds get caught up in unnecessary things despite our intelligence, and hence adopting the attitude of letting go and being non-judgmental in this practice will cultivate a mindful being in you. When you notice that your awareness is no longer on the breath, gently congratulate yourself. You have come back and are once more aware of your experience. You may also want to acknowledge briefly where the mind has been. Arey, mera mind abhi kahan tha? There is a thinking. So then gently escort the awareness back to a focus on the changing pattern of physical sensations in the lower abdomen. So try to escort the awareness back to a focus on breath. Notice that your thoughts and feelings, just notice your thoughts and feelings as if you are an outside observer watching what is happening in your mind. Do not try to label or judge them, and just gently allow yourself to return to your breathing. However, often you notice that the mind has just wandered, and this will quite likely happen over and over again. Here, you can also congratulate yourself each time on reconnecting with your experience in the present moment and gently again escorting the attention back to the breath. No need to try to control any of the thoughts. Just observe them as they are. These are just passing events. No need to react on them. No need to judge them. No need to label them, whether they are good or bad. These are just thoughts. Let them come as they are. And this is what the mind does. Here you can also bring a quality of openness to experience or kindness to your awareness. Don't try to be harsh. Try to be gentle to your awareness. This is just a practice. This is not a competition over here. Simply so bring kindness, gentleness to your awareness, perhaps seeing the repeated wanderings of the mind as opportunity to bring your patience and gentle curiosity to your experience. This might be a new opportunity for you. So you are putting on your efforts with gentleness, with kindness, with curiosity. So bring this attitude. Don't try to strain yourself. Continue with the practice for 2 to 3 minutes. Perhaps it might be reminding yourself from time to time that the intention of our practice is simply to be aware of your experience in each moment. That is why it is called moment to moment experience in the present moment. And here you can also assume the breath as an anchor, just to gently reconnect with the here and now each time when you notice that your mind has wandered and is no longer down in the abdomen following the breath. So assume the breath as an anchor. Bring your attention back to the breath. Let the thoughts come. They will come automatically and go automatically. No need to give any importance to these thoughts. These are not facts. These are just thoughts. These are just passing events. Again, try to notice the wandering nature of your mind. Just acknowledge them and try to return to what you had intended to be focusing on. If your mind is wandering a lot, simply observe the thought as passing events, then bring the mind gently back to the breath. Use your breath as a vehicle to help direct and sustain your attentional focus. Try to be in the moment, accept the distraction as they are, allow and accept the things as they are. So let go of rigid attitude. So by paying attention to a single focus, the breath will help you to recognize your old mental habits as they arise, most of the time. Don't try to label your efforts of focusing as success or failure. Let go of the attitude of labeling them as good or bad and success or failure. This is not a competition again. It is not a skill for which you need to strive. And also, let go of any expectations about this exercise will do for you. The only discipline involved in the exercise is regular and frequent practice of paying attention on purpose. Just do it with an attitude of openness and curiosity, then allow the rest to take care of itself. Gently bring the attention back to the breath. Now gently open your eyes.
And definitely it's going to help in all sense for our psychological peace of mind and everything, right? So definitely it's going to help. Thank you.
Okay, I have also got to say something. If it's working, is it? Okay. Even in such a structured room where we are having a very formal presentation, you know, everybody are in a little anxious state of mind. Even in such room, we can reach to such relaxation, you know, with a simple mindfulness. So imagine if you are sitting in your home in a very comfortable position, this mindfulness will be working 10 times more for all of you. So you don't need a very comfortable or a very, you know, cool environment near the nature. Anywhere, in your office, in your workplace where you are tensed, you can practice this and get all the benefits.
Restless mind, and ox mind refers to the calm and relaxed mind. So monkey mind is just jumping one thought to another thought. Earlier our thoughts were jumping one thought to another thought. This is called rumination. So through the practice of mindfulness, you can get a relaxed state like a ox mind, not monkey mind. It is monkey's nature to jump here and there. So similarly, our mind has the nature to jump here and there in terms of thoughts. Clear? Next slide. Now, which one will you choose? This is mindful of the things, and this is just mindfulness. This is your external experiences, and it's all about your needs and internal experiences. So which one would you like to choose? Which option? Second option. Yeah. See? So So thank you for your cooperation and attention. Thank you.
Thank you so much, Dr. Deepak. Now our next guest was Suresh Mehta. They have done MBBS and MD in psychiatry. They were professor and head of department of psychiatry PMCH. They have experience of more than 25 years. They were awarded as best intern and outstanding student. They were also felicitated by Teachers and Teachers Day. They were also an accredited national speaker, and they organized various conferences and workshops. They were also felicitated by Rajasthan Patrika for excellent work in medio-social field in 2022. They have more than 30 research paper publications. Now I would like to welcome Dr. Suresh Mehta to continue the session.
Hello. I would really appreciate the patience of all the members in the audience. This is the last session and I would like to request कि please आप लोग थोड़ा quiet रहिए, अपने phone switch off कर दीजिए. This will be the last session. So please आप patience थोड़ा और रखिए. We'll be free soon. Thank you so much.
Good afternoon, everyone. I am really sorry I am keeping you, all of you, hungry. Definitely that is also stress, but in life lots of stresses are there and we have to see for managing this stress. And that is the basic in the role of managing psychosomatic disorders. So initially it was a plan that understanding life skills and then activity, I have distributed, circulated those questions, papers, and we have talked about this, but because of time constraint, as it started 20 minutes late and as lunch is also waiting for all of us, we'll skip that activity. I'll continue with the lecture. So understanding life skills is very important because unless you have skills, you cannot tackle or manage the problem of stress. So life skills are defined as living skills or abilities of adaptive and positive behavior that enable individuals to deal effectively with the demands and challenges of life. And if they are there with you, definitely your outcome from the stress is in a positive way. So they are learnt over years, especially during childhood and adolescence. Every activity teaches you something. It's not that special classes are taken to teach you life skills. They come naturally, automatically from various things going on in your life, and that gives you lots of knowledge which you can use and tackle with other problems in your life. So life skills are used every moment of our lives in various situations, even example choosing friends, even taking decision that okay, Verma sir has requested me, invited me to take this workshop, and definitely I got motivated because of his push, and I thought, okay, I have those skills I can take. So all these things go together, and various life activities give you clue towards developing newer and newer skills. You never stop. You go on learning. It is said that you never grow old. You grow old only when you stop growing. So these skills will go through learning, will go throughout the life till the end of your life. Various skills are there. I will not go into those details. Critical thinking, creative thinking, decision making, problem solving, interpersonal relationship, effective communication, coping with emotions, coping with stress, self-awareness, empathy. Activity was there that we were going to ask these questions, that when to use which skill and what will help you in which situation. And that is the brain behind all the things which will decide which skills you will be using in what way. I am skipping this activity because it will take another 10 minutes. Sorry for that and sorry for the delay. So what is stress? As sir has given me sound foundation in his lecture about stress, so I will go into further details because unless you cope up with stress, you will not be healthy as sir taught you mindfulness exercise. Unless you are coping with all these stressors, all these things, it will have impact on your body, and mind affecting body leading to disease is a basic part or basic root cause of psychosomatic disorders. So this axis already sir has told you. See this newspaper headlines. Teacher shoots trustee in counseling meeting in California. Youngest CEO in corporate industry dies of massive heart attack. Student commits suicide in frustration. Major train accident in Spain. Train driver said he went blank under stress. Farmer commits suicides. So definitely it is alarming, and stress has so much impact on your body as well as mind and overall outcome. So in workplace, multitasking, most of you are overburdened with work. Always, ये करना है, वो करना है, this is that, like that. Unrealistic targets. You are having excessive, unexpected, unrealistic targets, so it creates pressure on your mind, and then it may disturb your appetite, it may disturb your sleep. Unhealthy competition. We unnecessarily go for competition and create more problems for us. Factors influencing work stress. This can be drive for success, always busy with I want success, should be successful. I can't tolerate failure in life. That is there. Changing work patterns, changing conditions, overwork, underwork, uncertainty, conflicts, responsibility, relationships at work, and change at work, favoritism, all these factors are there at workplace which will affect you. Everyone is different with unique perceptions of and reactions to events. Say, the approach or meaning which is taken out of some activities depending on the person's personality, his state of mind and everything. So everybody is different. How he has developed since childhood, in what situation he is at the present moment, all these factors will decide the overall outcome. So see this patient, it's the 25th doctor. He is seeing for his multiple complaints. Doctor is telling him it is hypochondriasis. He is asking, proof batao. I am a Google patient. I read lot of many things. Nothing is written like this. Proof though. I have done so many investigations. I have spent 2 lakh rupees and still I am not so happy. All doctors are useless. You are 25th one. So at least you give me this relief from the symptoms. So this may be the case in most of the cases. Most of the stress we experience is self-generated. How we perceive life, whether an event makes us feel threatened or stimulated, encouraged or discouraged, happy or sad, and that decides the outcome. I had 2 patients. One was student, say from our college, just for example. He has been brought by father saying that he is crying. He has got first class but crying that he doesn't get distinction. And another patient, student brought by father, see doctor, just try to, if possible, you can change his attitude. He has got 50%, he is happy, I saved my 6 months. I was expecting I will fail. So this is a difference in attitude of 2 students. Same thing, same exam, 2 students are having different approaches and outcome. That depends on the person. Emotions are there, which can be positive or negative. Positive emotions, happiness, love, frustration, satisfaction, pride, enthusiasm, elation. Negative are anger, depression, sadness, anxiety and frustration. Nowadays we come across lots of negative emotions. Everybody is saying, yaar, I am there इसलिए मैं जिंदा हूँ, नहीं तो कभी का suicide कर दिया होता, ये तो मैं हूँ. Everybody feels like that. So that is stressed out. They can be positive one are appropriate, constructive and helpful, while negative are inappropriate, destructive and unhelpful. Already discussed by Varade sir, personality traits. Depending on your personality, you will decide what will be the outcome. And attitude, belief and control will decide the further outcome. I was cursing God for my sore hands until I saw a man with no hands. There are many people who are more disturbed or more suffering than us. We just concentrate on our point, negative point. Whenever you are depressed, you are tense, you just try to look out who is more distressed than me. So you will feel happy, okay, मेरे जैसे दुनिया में बहुत लोग हैं. So that should be there. Mind traps, because we have unrealistic expectations, taking things personally, all or nothing thinking, exaggeration and rigid thinking. Poor socialization also leads to loss. As it is said that human being is a social animal. Okay. So if you are socialized, you are alone, staying for 1 month, 2 months, you get suffocated, you get frustrated, and that leads to lots of trauma or stress on your body. Emotional, feelings of loneliness, loss of self-respect and uselessness, all these factors will also affect. Emotional suffocation is there. It's like a pressure cooker. Sitti bajna jaruri hai. If it is not there, safety valve will go off. So that is ventilation which you need. Okay. Behavioral symptoms are there, which can be there. I will not go into all these symptom details. You can go and read afterwards if needed. There can be physical symptoms also, and they present to various doctors in the form of physical symptoms, but the root cause may be emotional factors which are leading to trauma, leading to disturbances, leading to physical symptoms. Situation 1, let's go fast with various situations. Mr. Sunil, 28-year-old person, parents forcing him to get married as nobody is there to look after them. Parents are anxious as there is an opportunity to go abroad for on-site assignment for 2 years with good salary. So whether he will go, leaving us alone. So he wants to go. But what about parents who are old and dependent on him? And what about marriage? He is in a dilemma since 2 months and getting severe headache with no relief. What to do? Whether he needs only analgesics or he needs something else. Yes, from audience please. सर ने आपको माइंडफुल एक्सरसाइज करवा के सुला दिया है शायद. Yes, anybody? Okay, so with giving analgesics, that is for time being it may give relief, but underlying root cause is his dilemma, not able to take proper decision and stress, which is leading to this. So unless psychological factors are looked into, he is not going to improve. Psychological symptoms, panic disorder, palpitations, chest pain, restlessness, sweating, dryness of mouth. So all these factors will also affect. Lack of concentration, memory lapses, difficulty making decisions, confusion, disorientation, fits of rage. There are so many symptoms which are presented, but underlying root cause may be psychological stress. So that's why I am insisting on these slides that psychosomatic disorders look into all these symptoms and the underlying root cause, that is important. Emotional symptoms, there can be depression, there can be bouts, there can be deterioration of personal hygiene and appearance, everything. So sad faces, easy to remember, symptoms of depression. A is for sleep disturbance, A for appetite is diminished or increased, D for dysphoria, A for fatigue, A for agitation or retardation, C for concentration disturbance, E for esteem, low self-esteem, S for suicidal ideation. Okay. So if you find these, easy to remember sad faces, if you find these, this can be indication of underlying depression. You take care of that, symptoms will automatically go away and patient may feel better. This is another case. Radha, 30 years old, working in software company since last 5 years, got married 2 years back, having 10 months old daughter, is feeling irritated for every small issue. Emotional outbursts have become quite common. She is also having multiple physical complaints. She doesn't have time for herself, feels she should take off, but she cannot for every practical reason. Boss is not allowing and she stays, "Main kya karu?" So anybody has solution for her? Vacation दे नहीं रहा ना बॉस, छोड़ दे, दूसरा जॉब मिल जाएगा, सिर सलामत तो पगड़ी 50. Okay, yes, you want? Yeah. So hope all these dreams can come true and possible. Otherwise, she may be having 2-3 things. One is 10 months old daughter, so there can be underlying postpartum depression because of hormonal changes. Secondly, previously she was doing same job, she was happy, but now additional responsibility of taking care of the child, that is also there. So all these factors have come together, led to lot of stress, leading to remote control, leading to effect on body, and leading to all these issues. Confused. Doctor be confused, patient be confused. Kya kare? What to do? So in such cases, taking detailed history, role play will cancel because time constraint. Already 10 minutes mein main finish karo. Thank you. Everybody is confused. Doctor is confused, patient is confused, family relatives are confused. They are saying, kuch nahi, drama kar raha hai, kuch nahi hua ise. Doctor is trying to kill. If he is cooperative, helping, supportive, then he will explain, educate the relatives about the disease. Relatives will say, यार ये इसने तो मेरी जिंदगी खराब कर दी, 25 डॉक्टर हो गए, 5 लाख खर्चा हो गया, ये नहीं होता तो अच्छा था, इसका हेडेक हो रहा है, मुझे हेडेक हो गया है, मेरी जिंदगी में हेडेक बन गया है. So that will be the attitude and that will lead to worsening of the condition. So stress has all these factors, behavioral, physiological, cognitive and emotional, and all these factors will affect each other, leading to disturbances. So unless all of them are stable, you cannot be physically or mentally also stable. So this is a vicious cycle and each factor affects another, and that's why you have to take care of all the factors, and that's why role of stress management is there in lots of psychosomatic diseases. So there can be disease in any system of the body because of stress. Okay, I will not go into all the details. So ask the right questions. As a doctor, as a colleague, as a room partner, as a friend, you can ask these questions. Have you been under stress recently? Have you been worrying about anything recently? Have you been feeling unhappy about anything? Do you at times feel as if you have had enough of life, want to end your life? And do you at times feel scared or tense? So if there is yes for any question, that indicates there is underlying stress, and unless you take care of that, patient will not improve. End हो गया? इतना जल्दी खाना खाने नहीं छोड़ेंगे. Silence speaks a lot. Many times silence is there which you have to understand. Patient is not speaking. Many times it happens, mother-in-law has brought daughter-in-law with multiple complaints. You are asking, सब तो ठीक है? She is looking at mother-in-law. That indicates is nothing is stick. There is some problem in the relation, and unless that is settled down, she is not going to improve. So, please look for hidden messages and body language. You have to read between lines, and that will give you clue towards that. We'll skip role plays because of time constraint. Attitude should be like this. This is what we call as healing touch. Healing touch should be there. You should treat the patient with empathy, with sympathy, and then patient will recover in a better way. Mother Teresa. Being with her, this child is also doing, this child is also doing like this. So you have to learn from others, learn from their body language. So it is much more important to know what sort of a patient has a disease than what sort of disease a patient has. Okay, that is very important, that unless you understand the basic psyche or personality of the person, you are not able to understand or treat the disease properly. So that is there, that is very important. Many times it happens that patients will tell you, डॉक्टर साहब आपने जिस तरह से बात की, आज तक 20 डॉक्टर्स के पास में गया और किसी ने मुझे इतना पूछा नहीं, किसी ने मेरी इतनी जानकारी नहीं ली. No, I feel I am in the right hand and I will feel better. So that should be the comment from patients when they come to you. So kinds of problems people face: relationships, employment, finances, housing, social isolation, use of alcohol and drugs, physical health, sexual adjustment, bereavement, and legal aspects. All these stresses can lead to impact on your body. This is 8-year-old girl is getting recurrent pain in abdomen since 3 months associated with at times vomiting and not eating properly. Her academic performance has deteriorated and teacher reports poor concentration in class with at times absenteeism. Investigation reports are fairly normal, physical investigations are fairly normal except gastritis. There is recent history of change of school and separation of parents due to frequent fights and physical abuse. So what is needed to treat this child? Settling down of her emotional issues rather than just looking after vomiting. So such history is there, you have to pay attention to all these factors. So brain has so much to do, so he needs relaxation also and regular exercise to make it fit. 5 minutes. I am rushing through the slides. Maybe most of you may not understand so fast. I hope you can pick up some points. Expression of work-life balance was first used in 1986 in US to help explain unhealthy life choices many people were making. That is it. So, the basic part of treating psychosomatic disorders is change in thinking, change in behavior, and change in lifestyle. Whatever is possible, that will definitely give you better results. Stress assessment questionnaires are there which can be utilized. Illness model is important. There are various cognitive pictures which should be assessed. Illness model, personalized formulation, affective disturbances, motivation, coping, life transitions, influence of others, and resilience. So role of counseling makes a lot of difference, and there are, I will not go into other details, but there are lots of techniques like relaxation technique, progressive muscle relaxation technique, same continued. So, see, this is role of family member. In monkeys, they stay together, they have support of each other. Nowadays, because of nuclear families, this support has gone. Ventilation, vent out, speak out. कोई तो चाहिए सुनने के लिए. सुनने वाला ही कोई नहीं है. पैसा बहुत है, आदमी नहीं है. सुनने वाला नहीं है. That is main point. Cognitive behavior therapy which changes your behavior patterns, modulation. So in 5 minutes I will just come for basics. It's okay? So where I am now in this? I am just going on giving lecture. All of you are listening. Anytime you have tried to know where I am in all this, what is my present status, whether I am stressed, I am healthy. Why do I want to work? What do I get out of it? What do I want from my life? Do I have life other than work? How is my support system, and do I really understand myself? Ask these questions and you will understand where you are and whether you need some help. Not psychiatrist always, it can be friend also. Do I want to pay this cost? Ask yourself. What do I know about myself? How do I look at myself? Am I missing myself? Ask yourself. Spend 5 minutes for yourself. Work is going to be continued. As he mentioned, छुट्टी ले लो, दूसरा job देख लेंगे. So most of the times we have denial, we have rationalization. नहीं ये तो ऐसे ही चलेगा, ठीक है. Burden of past, prejudices, and rigidity of thinking. So let's change something. Decide today, to determine today that we will change something so that we will not need Dr. Mehta, Dr. Varade or Deepak sir. Okay? So be positive, have self-assessment, correctly judge your capabilities, rectify your deficiencies, self-motivation attitude, and never give up. Practice learning. जिंदगी भर सीखते रहो, अच्छी चीज है. चेंज तो एक्सेप्टेड है और टाइम का पार्ट है. लिसन टू योर बॉडी, लिसन टू योर फीलिंग्स, लिसन टू योर स्पिरिट, लिसन टू योर रिलेशनशिप्स. दे विल गिव हिंट. आजकल ज्यादा गुस्सा कर रहे हो, आजकल बहुत ही ज्यादा मस्ती है. यू आर गिविंग इनडायरेक्ट हिंट्स दैट यू कैन पिक अप. Eat wisely, consult your physician or psychologist, exercise, take time to rest, relax and recreate, set goals in life, adapt to change, communicate your feelings and think positively. Good food. ऐसे खाना है, ऐसे नहीं खाना है. किलर सोफा, रिलैक्स होके सब फास्ट फूड खा रहे हो, एंजॉय कर रहे हो प्रोग्राम, बाद में डायबिटीज ये वो सब शुरू हो रहा है. एक्सरसाइज, it increases, lots of benefits are there. You should have 15 to 20 minutes warm up or 20 to 30 minutes moderate exercise twice a week. ये relaxation नहीं है. But most of us go for this. अरे यार टेंशन आ गया, चलो मोबाइल देखते हैं. देखो ये बच्चा क्या कर रहा है, आगे की स्लाइड ध्यान से देखो. भगवान मेरी मम्मी का व्हाट्सएप बंद कर दे, नहाने के बाद 1 घंटे से नंगा घूम रहा हूं. So this not, I hope this doesn't happen in at your place. And you should not be so addicted to mobiles. That is worst addiction at present day. You should be so flexible to relax in any position. Go for yoga. Get organized. Time management kijiye. I am sorry today we could not manage the time because it started late and all of us are hungry waiting for lunch. Relaxation activities, recreation, humor. हंसना जरूरी है. वो भी ऑफिस में बैठे के ऐसे नहीं, दिल खोल के हंसो, कोई प्रॉब्लम नहीं है. ऑल मसल्स शुड वर्क एंड रिलैक्स. फाइंड समवन टू गिगल, नॉट गूगल. व्हेन लाइफ स्टार्ट्स फीलिंग अ बिट टू सीरियस, फाइंड समवन यू कैन गिगल विथ. 3A's 1BC, alter it, avoid it or accept it. Build your own resistance, strength बनाओ and change our perception. I'll skip these slides. So just, most of us say, just do nothing. कोई फर्क नहीं, जिंदगी चलते रहेगी, सालों 100 साल जीना है, क्या गड़बड़ है? Just do nothing. Others will say, it's impossible. आप कुछ भी करो, फेल नहीं होने वाले हो, पास नहीं होने वाले. Impossible. Just join two and see what happens. Just do it. Nothing is impossible. So plan and go ahead for doing something. And if you use all these techniques, events and coping, rather than getting stressed, just reverse the spelling, what it happens? Desserts. So now decide in lunch you want to enjoy desserts or no. Or you want to get stressed out. Spare some time for you and be childish like this. Enjoy your life. Relax. So you will never need all of us. I ask God to give me everything so that I can enjoy life. अरे यार उसके पास मर्सिडीज है, उसका तो इतना बड़ा बंगला है, वो तो सबके लाडली है, वो तो पास ही होने वाली. So I ask God to give me everything so that I can enjoy life. God replied, I have given you life to enjoy everything. So leave it up. Decide now to enjoy life and have such smile in your life. Okay? So special thanks to all my patients who taught me a lot and polished my skills. Thank you. Cancel kar do. If anybody has this.
House is open for the questions if anyone have. We're spending 5 minutes for the question and answer session. No one? Yes, sir, we all are relaxed.
Yeah. Does anybody have any question regarding meditation, mindfulness, or mind dullness? No question? Thank you. Thank you very much.
Thank you for being here.
Now they are not reacting. They are completely mindful now. Thank you everyone.
Thank you Deepak sir, Mahindra sir and Mukesh sir. Mehta sir, Mehta sir. Now I would like to request everyone to join for lunch, and after that we will again gather here for valedictory program. I would like to request Dr. Prachi Vyas to please felicitate our guest, Dr. Mahendra. I would like to request Dr. Prachi to please felicitate Dr. Mehta.
switch off your mobile, or at least put it in silent mode so that there's no disturbance during the valedictory. I request audience to kindly be seated. Please put your mobile on silent mode. Khambagani. Exploring boundaries, igniting minds. Science unites us all. Dear all, I, Dr. Mansi Verma, Joint Organizing Secretary of ICBTM 2024, welcome you all for the 3rd International Conference on Integrative Chemistry, Biology and Translational Medicine, organized by Hansraj College, University of Delhi, Pacific University, Udaipur, and Hetero Biochem Innotech, New Delhi, Bharat, in exemplary partnership with world's renowned institutes including Kumaon University, Nainital, Bharat, University of Debrecen, Hungary, Oswaldo Cruz Foundation, Brazil, Charles University, Czech Republic, Federal Rural University of Pernambuco, Brazil, Institute of Eminence, University of Delhi, Bharat, Mayo Clinic, Florida, USA, Miranda House, University of Delhi, Bharat, Jawaharlal Nehru University, New Delhi, Bharat. These past 3 days have been an incredible journey filled with enriching experiences. We have been privileged to hear from insightful speakers, witnessed groundbreaking research through captivating posters and oral presentations, and immersed ourselves in the scientific aura that permeated every moment. So, to start the valedictory event, I would like to show our gratitude to Lord Saraswati by lamp lighting. For this, I would like to invite our esteemed dignitaries, our chief guest, Dr. G. Satish Reddy, scientific advisor to the Raksha Mantri and ex-secretary DRDO, Professor Lesker Julian, MIT Smart, Singapore, Dr. Brajesh Rathi, convener ICBTM 2024, Shri Hemant Kothari ji, Dean PG, Pacific University, Udaipur, Professor K.K. Dave, President Paher, Udaipur, and Shri Sharad Kothari ji, CEO, Pacific Group of Education, to please come on the stage to light the lamp.
या कुन्देन्दु तुषारहार धवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा शुक्लां ब्रह्मविचारसारपरमाम् आद्यां जगद्व्यापिनीं वीणापुस्तकधारिणीम् अभयदाम् जाड्यान्धकारापहाम् हस्ते स्फाटिकमालिकां विदधतीं पद्मा.
Dignitaries to take their seat on the dais. In light of this joyous occasion and our collective success, it is my honor to extend a warm welcome to Dr. G. Satish Reddy, our chief guest, and for this I request Professor K.K. Dave to felicitate our chief guest by presenting Uparna and Pagri. Now I request Brajesh Rathi to present memento and bouquet to our chief guest. I request Dr. Lokesh Chandra Mishra from Hansraj College to felicitate Shri Sharad Kothari ji by presenting Upanna, Pagdi, memento and bouquet. Moving further, I take privilege to invite Dr. Brajesh Rathi, the convener and the driving force of this esteemed event, to present the report of ICBTM 3.0.
A very good afternoon to everyone. Our respected Dr. G. Satish Reddy ji, chief guest of today's validity program of ICBTM 3.0. Mr. Sharad Kothari ji, Professor Dave ji, Professor Julian Lesker, and Dr. Hemant Reddy ji. Kothari ji, sorry. A warm welcome to each of you as we gather for ICBTM validity session in this afternoon, and I'm deeply honored to express my gratitude to all of you for joining us today. The event showcased a remarkable lineup of presentations, workshops, panel discussions, and each offering the valuable insights and perspectives to everyone. Renowned speakers shared the groundbreaking research findings, innovative methodologies, and thought-provoking ideas, igniting intellectual curiosity and sparking meaningful exchanges among attendees. In addition to the intellectual simulation, we have had the pleasure of exploring the enhancing city of Udaipur, known as the city of lakes, and indulging in its vibrant culture events. Behind the resounding success of this event lie the invaluable contributions of esteemed institutions involved: Pacific University Udaipur, Hansraj College University of Delhi, and Hitrochem Innotech Private Limited. At the outset of the event, our anticipation was set for just maximum 500 participants. However, to our pleasant surprise, the enthusiasm and interest far surpassed our initial expectations, with the attendance soaring to over 800 participants. And this remarkable turnout speaks the volume about the significance and appeal of our event, underscoring its relevance and impact within the scientific community, in particular when we talk about interdisciplinary sciences. Our scientific event was featured in prominent media outlets, including Dainik Bhaskar, Rashtradoot, Agamata Media, Dainik E-Paper in Udaipur. We extend our sincere thanks to everyone around the world who has joined us and shared their innovative ideas for shaping up the future. One nation, one health. Our deepest gratitude goes out to all of our collaborators for their invaluable contributions, co-partners, University of Debrecen, Hungary, Kuma University, Nainital, Fiocruz, Brazil, Charles University, Czech Republic, Federal Rural University of Pernambuco, Recife, Brazil, Mayo Clinic, Florida, Miranda House, University of Delhi, Jawaharlal Nehru University, Bharat. As we conclude this remarkable scientific event, let's carry forward the spirit of collaboration and innovation. May the connection made and ideas exchange continue to inspire us. With gratitude to all who contributed, let's move forward with renewed enthusiasm. Together, let's arrive for new frontiers of discovery and advancement, enriching our future with knowledge and understanding. Until we meet again, let's keep pushing boundaries and making strides in our scientific journey. I'm really glad to share that our next ICBTM, that is going to be the fourth edition, and that has been announced officially, and that will held in Brazil. And for this, I would request Dr. Pena to come and say a few words about that, and then we can continue with the session. Thank you very much.
Good afternoon, everyone. Namaste. So we are very happy to be here. We want to thank the organizing committee for having us. This is the third conference. It's the second that we have come from Brazil. We want to a special thanks to Dr. Ratti. He is the one that is the catalyst of this reaction. Without him, we'll not be here. We are very happy with him because besides being an excellent collaborator, he has become a friend. He's going to be in June in Brazil. And I told him, I don't want you to stay in a hotel. You stay in my house in Brazil. So we are family. And we are very happy to have this conference in Brazil. We live in the city of Recife. We do have very good infrastructure. We do good research, good teaching, good training. So you are all invited to come down to Brazil, enjoy our beaches, our cultural heritage of Recife. It's a unique city, not the only in Brazil, it's a unique city in the world. If you go to Recife, it's different from the rest of Brazil. It has a very unique culture, unique dance, unique songs, so you enjoy. We are looking forward to see you in Brazil. Dhanyavad.
Thank you, Dr. Lindomer. I extend a warm welcome to our chief guest, Dr. G. Satish Reddy, the scientific advisor to the Raksha Mantri and a globally recognized defense scientist who has spearheaded the development of key defense technologies and equipment. His contributions have earned him numerous awards and honorary doctorates from prestigious national and international bodies. Notably, he is the first Indian in over a century to receive an honorary fellowship and silver medal from the Royal Aeronautical Society London, and the first non-American in nearly 40 years to win the Missile System Award by the American Institute of Aeronautics and Astronautics. His accolades also include the full foreign member from the Academy of Navigation and Motivation Control, Russia, National Systems Gold Medal, National Design Award, IEEE Award for Engineering Excellence, the Homi J. Bhabha Gold Medal, and a Lifetime Achievement Award from ISSE. Additionally, 24 universities have awarded him honoris causa degrees. Wow. Big round of applause. I request Sir to address the gathering. Welcome, sir.
Honorable Vice Chancellor of Pacific University, Professor Dave, Professor Rathi, who is instrumental in this conference, then CEO of the Medical University, Shri Sharad Kothari ji, Shri Hemant Kothari, and our friend Professor from Singapore, Mr. Leskar, who is here from Singapore University, and all the other eminent personalities, professors, researchers who have come from many countries abroad and also from the soil of Bharat, and most importantly the youngsters and students who are present here. Very good afternoon to all of you once again. Let me thank the organizers for inviting me here to be on this occasion of valedictory event of the International Conference on Integrative Chemistry, Biology and Translational Medicine. Last time it was in Delhi, the conference, wherein I was there in the inaugural along with our Honorable Minister for Jal Shakti, Government of India, Shekhawat ji, and we were together and we were very happy to be part of the conference, and also we were excited about the theme of the conference also. Why I am saying this is slowly the days are going where you have a very specific subject, structured way, and working in that particular area and teaching in that area, getting a degree in that particular area. That concept is slowly moving away with the type of opportunities, with the types of subjects which are evolving and the types of interdisciplinary subjects which are evolving and all that. So the concept is changing. Even if you look at our country now, the new education policy and the national credits framework, what has been worked by the UGC, if you look at the credits what a student need to have, whether it is 160 or 120, 140, in that what is in the core discipline, what are in the other disciplines, they all have been well defined, where people are trying to work on multiple domains which are connected to it and trying to have credits on various courses. Probably if you look at the last 2 days, Professor Jagadish, the chairman of the University Grants Commission, has written 2 articles. If I'm right, yesterday it was in Economic Times and day before yesterday on the same subject, how a particular degree has been given by a university and what was the earlier framework of teaching, curriculum, what it used to be. Now the way the evolution is happening in various disciplines and the requirements from the industry point of view or the society point of view, how various other subjects can be brought in, and freedom is given to the student to choose the various courses and credits and come out of it, has been the theme of that. And in fact I was just trying to go through, I couldn't go through it today, the total framework of the national credits framework, which was trying to, probably many of you would have gone through and trying to get into it also. So that is one of the important aspects which has started happening. I think the conference is also in the similar lines, but I don't want to exactly relate to it, but in the similar direction. Most importantly here, where the subjects of the biology, the chemistry and translational medicine, which are very much interlinked with each other, and trying to, anyone who is working in this should have a reasonable good knowledge in the all other subjects is what is very, very, very important today. If you look at till recently, till now, a person who is working in the pharmaceutical industry in the research is too much into that specifics and working in that chemistry and chemistry related things and coming out without much of the details about other subjects at all. And similarly, if you look at a doctor also, probably he has got in the first year or something like that, some subjects are taught, but then not much having knowledge into the pharmaceutical field, what type of a chemistry is involved into it and what are the various ingredients and how it is and what is the effect of it and this. Similarly, if you look at probably over years, maybe decades or centuries, looking at the body as a whole, taking the genetics into consideration or various aspects into the consideration, that sort of a thing in the medical field also has gone. What I am saying is, if you look at the Indian knowledge, the body, mind and everything together has been seen as an integral part of it, and then when you look at a person, whatever the medical, various medical fields what have been there in the Indian subjects, or maybe many other areas also, many other countries also who have been following the traditional methods which have come out from that soil, they have been looking as an integral entity, as that's the concept that has been there. You just look at it in the Indian way of, I think the most important was the Nadi Shastra. Touch the Nadi and look at the whole body as one, and looking at the facial part of it, looking at the tongue and various things people used to tell and say and all that, and come out with a very, very detailed way, looking at any part of the body or anything you have a issue, people used to come out. In fact, let me tell you, I got interested into it a little bit when I saw an instrument developed by Russia, the northmost part of Russia, an instrument which actually works on this principle and generates about 108 pages of a report of any human being once you connect it. The name of that instrument is put as Veda Pulse. It is called Veda Pulse. There is an instrument, you can Google it and also and see that. I myself tested it and got it. These people somewhere in 1980s, when they interacted with the Indian doctors here and who have been experts in Nadi, they took the knowledge and then developed this instrument and made that type of a thing, and which is good. Actually, I have seen through it and gone through it and all that. So that's where actually one of the important things where Indian system was looking everything into an integrated way. Even now there are many people who actually do the study like this and tell and all that. For your information, last 6-7 years I also get just checked and get rather treatment, whichever way you call it as, by one famous Nadi Vaidya in the country. He looks into it and he tells me a small fluctuation in the BP and sugar also, or anything else also, and a very, very, very small remedies and all that. Looking into every aspect of it, including some of the medicines what need to be given. He actually says natural medicines or Ayurvedic medicines, and sometimes suggests other things also, or yoga related, or also even English medicines also for some particular applications and all that, whatever we call it, allopathic medicines, even that also suggests. So that is how it is. What is important about the conference, and when we say it is looking at the constituent elements or constituent subjects as an integral one, having a knowledge of everything and trying to treat a human being and try to develop a medicine, looking at various aspects is one of the important things. And in fact, what happens is, probably you are all done so much of work into each and every area, and you are all the experts and all that. But what, from my point of view, what I have studied and all that, or gone through, the every human being is actually is a specific identity, has an identity. Every human being has got, and place to place, country to country, or in the country across various places, many things are different. If you look at the various data of the genetic engineering, the number of different kinds of this and data what it has, if you look at it, is there are so many changes and so many differences between across the countries, across the people, across the places, and all that you will be able to see. And also it is all because of, one is the origin from where he has come and what is his background and all that, and the place where he is living, what type of environment he is going through, and what is the type of food habits he has got, and based on that, these are all differentiated or classified and all that. One of this person only was telling, he has classified into 768 types of it. And then when you look at giving a medicine to someone, he looks into all these classes and then try to give the medicine in what type he fits into it and all that. That is why these days, if you look at it in the modern science also, the personalized medication is one thing which people are talking about, looking at each individual, what type of the structure he has, and based on that you try to generate your own medicine and give, is what people have been talking and people are working. Other day also somewhere in another conference, somebody was working on it, and then he has developed AI based, and with the database what he has got over the country and particularly in India what we have, and using that trying to develop the medicine and give it to it is one thing what many people are working. I think a lot of startups have come up here in the country who are also working on this and trying to work in a way, looking at the genetics, looking at the person, looking his own habits, and looking at the way he has been living and type of food he is taking, and then so you try to develop the particular medicine and try to give, develop not now, you actually composition of the medicine, you give it to that person, is what people are working on it. So that is why last year I was telling you, now also looking at the theme of the conference is very important, very apt. Probably maybe in couple of years, one or two more subjects also may join into it, and which you will integrate into it and try to say not just chemistry, biology, and translational medicine, and maybe the food related things and naturopathy related things and all that. Probably you will be joining it also, maybe happening, which is important. Actually, that we didn't have this concept of some time back, that too much specialization into one thing also. It is nothing wrong to be in too much specialization, but having a wide knowledge of various things and try to have with that knowledge base and a specialization is what is very important. In fact, let me come back to my, not medicine related, even in engineering also. 40-50 years back, the types of subjects what were there in the first year and second year, now they all have vanished. You are all trying to focus only on very specifics right from the first year, and you do not know anything about basics of other engineering aspects or physics or chemistry part of it and all that. Whereas when we studied, leaving aside 11th and 12th, even in engineering, we were studying physics, we were studying chemistry, we were studying various other subjects. Even an electronics engineer was studying the mechanical engineering subjects also in almost for the first 2 years, and all that was the type of a concept what was there in the country, which actually vanished. Probably through these credits and electives process, now again we are getting into it and trying to look at various subjects and try to see. I'm happy that today this conference, as Professor Rathi was telling, was attended by more than about 850 people, and I think about 53 experts have given their talks over the 3 days. and there are more than about 8 countries or so people from various places have come and participated. A lot of international presence is also seen here. And then also was told that youngsters have participated in it in a big way. More than about 150 or 160 youngsters, paper presentations and various other things is a very good sign what we are seeing. It is in fact actually very important the way the youngsters are evolving in the country or elsewhere also, if you see, in every field, including chemistry, including medical field, is a lot the way the number of startups which have come up in each and every area are too many. Too many means I am not saying that they should not be that many. I am saying that they have evolved themselves in such a way that they are coming out with most modern techniques and mechanisms and both hardware, software related and all that. In this country, in the year 2016, we had just 460 startups. Now today the latest number of startups in this country is about 1,14,000 startups. 1,14,000 startups. And you look at, I've been interacting last about 3 to 4 months, many startups. I'll just talk about only medical related. I went to ISB. So somebody developed an app with image processing, with a mobile camera, and look at into the eye. Cataract is wise. 96% success rate. Government of India has recognized it, and in the Northeast, more than about 1 million people are being tested using that young boy, about 25, 26 years old. Then one more in the same ISB, Hyderabad, again taking the mobile camera and then looking at the mouth and looking at the various ulcers and all that, and coming out with probability of a mouth cancer or even deep related to these, whatever is connected to it, whether it will be turning out and what is the time period and all that. It's considered to be one of the very good apps which is being internationally evaluated now today. That's another important. Then I found another boy, SMA, shaped memory alloy based, a small material insert into the body and then to close the holes in the heart, sends it inside and then gives the pulse. It opens up there and then closes the hole. In fact, that has been experimented on 5 patients, which is incorporated and working also, working very well. So likewise, if you look, there are many things actually. You breathe out and then analyze it, and you are able to say most of the parameters, particularly connected to the kidney and related to the lung, various aspects, including even blood sugar levels. Very close to realizing the product. The product-wise, people are able to come out with this type of products. This is just not possible with working in one particular subject and not knowing anything and all that. This is where you need to have the physics, the chemistry, the sensors, the detectors, and then the knowledge about anatomy of the body and body related all health parameters, related things, and then electronics and then software is what could have made these people work. Just 3-4 people who have worked on that and made this type of devices. I'm just giving you glimpses of some of those small things. In fact, if I am right, last 3-4 months I would have gone at least about 30 to 40 startup such instruments which are coming up, what I have seen in recent times. In fact, 15 days back, Boeing has conducted one competition. In that, it's called a BUILD program, BUILD, Boeing University Innovation Leadership Development Program or something like that. All over the country they have conducted analysis and invited a lot of startups to come forward, and about 2,000 things startups have participated and evaluated. They came out with only about 28 in the end, and 7 of them have been given a lot of funding and awards and all that. In that 28, there have been about 7 connected to the medical with the interdisciplinary subjects of chemistry and biology related things. I was astonished at the type of innovations they have come out with various things. One was connected to vision related thing, and one was connected to the cochlear implant related thing for the hearing and all that, and so many things also. Some normal things based on the vision and other things have also come out, and that's a separate issue. But this is the type of things which are happening. So, in this scenario, in this situation, and the way the things are evolving today, both environmentally, the living styles wise, and whatnot and all that, it's very important to have connected interdisciplinary studies, research, and knowledge is one of the important things. And so I appreciate definitely Hansraj College for taking the lead and having the conference last year, and now here also with the Pacific University and then Heterochem Innotech together along with many other universities including JNU here in India and various other universities abroad for taking this subject and going through. Here one more important thing I like to say that you have gone through 3 days intense discussions, and people who are experts in this each domain. I've gone through some of those things, various proteins and protein related studies onwards, many things which people have talked about it. End of this, what is that we need to do also, please look at it. We have come out in the discussions, or probably each session in the end you must have had some discussions and you would have come out with what should we do next. Either as this team of who is holding the conferences across the globe, or otherwise as a nation, as a globally for anyone, what is the way forward in this also we should be able to come out. And let's say for this country, having gone through it, can we give a recommendation to either Ministry of Higher Education or to the UGC? This is what we have thought about, this is what experts have said, and so let us have some courses like this, and let us have a curriculum like this, and let us introduce these subjects into various degree courses which are right now structured as degree courses. These should be introduced. Sort of a recommendation, my feeling is, should go through. If you want, I can help in that direction also. Both I can get you an appointment for UGC chairman. Both of them are my good friends, Secretary Higher Education, the Chairman UGC, Mr. Professor Jagadish. Both of them I can talk to them and see that this can be incorporated in that. For that, I think a small team after this need to sit together. This is what we have discussed, and this is what the conference, and talk about the specific recommendations what you are giving to be incorporated. If someone incorporates, probably it will be done also. Next year, our friend in Brazil also, they will also give a recommendation and try to incorporate in that country, and then globally it becomes that people will be having in every country like this. Is one thing what I think you should definitely do, because any event you do, there should be a purpose, and purpose should not be that we have discussed and all of us met and then gone, but after that there should be an action and a roadmap to it. That's what I feel. I can help in this direction. So, my best wishes to all of you, and my best wishes to my friend from Brazil. That is, when is going to be that conference? Next year. 26. Yeah, so 2 years. 2 years. Very good. So I wish all the best to you. I wish that there will be a lot of participation from all over the globe. I can see from last year to this year the change within the country and also global participation also. I wish that it expands in a big way and you will be able to give a message to all the people. And I really appreciate the Pacific University for hosting this conference here. And probably you also have put in a lot of efforts to see that many people are able to participate. so good. I appreciate and congratulate all the people from the Pacific University for this, and my best wishes to all of you also. And best wishes to the friends who have come from abroad all the way, and I wish you had a good stay, and this is one of the most beautiful places. And I don't know whether you know or not, Udaipur, there is a concept in India which has come up, is called destination marriages. And so people go away from your place and somewhere else you get married, not that. So that this is one of the most popular place in the country for a destination marriage. So probably you can also think you can have some destination marriages from your country coming here and having in Udaipur. You can see that. So very good and all the best. And once again, please see the importance of this theme and try to work in this direction. Try to expand it also some more areas and give the recommendations so that the government of India can implement it. My best wishes to you. Jai Hind, Namaskar.
Thank you so much, sir, for your inspiring words and your vision for interdisciplinarity. Nadi Shastra is a true example of advanced Indian science during that time, and we are heading in right direction with more than 140,000 startups and research innovations. So I would again like to thank you, sir, for being with us at this platform. It gives me immense pleasure to announce that letter of intent has already been signed with Hansraj College, University of Delhi, Pacific University, Udaipur, University of Debrecen, Hungary, and Fiocruz, Brazil. A big round of applause. Now, I would like to invite Shri Hemant Kothari ji to kindly address the gathering with words of appreciation. Welcome, sir.
A very good afternoon to one and all present here. ICBTM 2024, an international conference being jointly organized by Hansraj College, Delhi University, Delhi, Pacific University, Udaipur, and Heterochem Innotech Private Limited, and co-organized by Charles University, Czech Republic, Durban University of Technology, Durban, Federal University, Brazil, Fiocruz, Recife, Brazil, Jawaharlal Nehru University, Delhi, KwaZulu Natal University, Durban, Kumayu University, Nainital, Loyola University, Chicago, USA, Mayo Clinic, Florida, USA, Miranda House, University of Debrecen, Hungary, is successfully concluded. It was carried out for 3 days with great vigor and quest to share and explore something new which can benefit the global society as a whole, especially in the era of post-COVID, where most people either themselves or some of their beloved ones are suffering from some of the other kind of health issues. It's my strong belief that this international conference, which has witnessed by 800 participants and with more than 200 scientific presentations by our delegates and more than 50 expert scientific talks, would have definitely paved new thinking and solution to the many emerging health paradigms. With these words, I compliment the winners for their outstanding scientific presentations and research work. I also wish all the participants of ICBTM 2024 a very bright future. Respected dignitaries on the dais, delegates, heads of institutes, faculty members, dear participants. With all due grace and dignity, I, on behalf of organizers of ICBTM 2024, extend heartfelt thanks to our humble, very kind, generous chief guest, Dr. G. Satish Reddy Sahib, for accepting our invitation and sparing his valuable time to be with us all today. Sir, it's my strong feeling that your words would have definitely invoked new light and thought among the august gathering and would have motivated young students to do something good for the nation. In absentia, we are grateful to our Honorable Chairperson, Shri Rahulji Agarwal, for his unprecedented support throughout the organization of this mega international conference and always thinking for extending each and every comfort to the participant and giving us liberty to take all the initiatives towards this pursuit. We are also grateful to our invited speakers for their thought-provoking scientific discourses and taking all the pain to travel to Udaipur from different continents of the globe. It was your presence and exemplary presentations which turned this conference into an unbelievable magnitude and has taken it to new heights in scale. We are also indebted to Honorable Vice Chancellor of Delhi University, Professor Yogesh Singh Ji, Professor Rama, Professor Balram Pani, Dr. Premada Singh, Dr. Mansi Verma, and other colleagues from University of Delhi for their wholehearted support. I compliment, thank, and acknowledge Professor Bijesh Rathi that it is his hard work that we have been able to hold and conclude this mega international conference here with exemplary expert lectures, technical sessions, workshops, and plenary sessions, and wish him all the best for his next ICBTM conference at Brazil. Success of any event depends upon constant encouragement, support, and engagement. And for all this, I sincerely thank Shri Sharatji Kothari for such humble cooperation despite his very busy schedule since the early days of planning of ICBTM 2024. I honestly thank our President, Honorable Professor K.K. Daveji, for his constant humble support in organizing this conference and backing all of us day and night, and even gone out of the way on many occasions to help us that this conference can achieve grand success. It's my proud privilege to acknowledge our co-organizers and knowledge partners for their technical and non-technical support in this academic pursuit. Looking to the limitation of time, it's difficult to name each and every individual. But from bottom of my heart and on behalf of all the organizers, it's my honor to accord special thanks to all the advisory committee members, invited guests, judges, session chair, co-chairs, rapporteurs, moderators, and local organizing committee members with Professor Dilendra Hiran, Professor P.K. Chaudhary, Professor Vinod Bhaduria, Professor Anurag Mehta, Professor Rameshwar Ameta, and their other fellow committee associates such as Registration Committee, Transportation, Food, IT, Technical, Press and Media, Accommodation, Discipline, Stage, and so on, for sparing and sharing their valuable time, relentless support, untiring efforts, and motivation towards smooth conduct of this conference. Last but not least, I thank all the participants and delegates from both within the country and outside the country for their scientific contribution in this conference, and traveling from across the globe to present their research and making this conference a grand success. Thus, due to everyone's efforts and participation, it has been an unforgettable experience, and I hope everyone takes away some wonderful memories. Finally, I thank you all again, and may all we be blessed with happiness and joy. Jai Hind, Jai Bharat.
Thank you so much, sir. With the crowd that big, it's time to roll out the red carpet and reward amazing students and participants with some well-deserved awards. And I request all the dignitaries to do the honors to motivate all. All set to go? Ready? So for poster presentation, the winners are Neha Agrawal from Paher, Udaipur. तालियों की आवाज गूंजती रहनी चाहिए. पर्व गुप्ता, RUHS CMS Jaipur. Anshika Sharma and Harshita Roy, Miranda House. Urvi Dave, BN College of Pharmacy, Udaipur. Ati Pujya Jain, Hansraj College, University of Delhi. Vidhi Upadhyay, Kumaon University, Nainital. Shubhashri Mondal, Kumaon University, Nainital. Lipsa Sahu, Hansraj College. Mani Shankar Kumar, UPES, Dehradun. Soumya Ranjan Pradhan, ICMR and NIMR, New Delhi. Anjali Miranda House. Indrani Chakrabarti, KIIT University, Lucknow. Aditya Arora, Department of Chemistry, University of Delhi. Deepit Paliwal, Paher, Udaipur. Anjita Miranda House. Congratulations to all the winners of poster presentation. Now it's time to announce awards for oral presentations. Time for oral presentation awards. So, excited? Himadri Shukla, CSIR CTRI. Anshuman Chandra, JNU. Padam Raj Bhatt, HRC. Tamanna Rajpal and Dr. Maya Sharma, Paher, Udaipur. Talia. Rahul Kumar and Jayaveer Malik, Department of Chemistry. Plabita Paul, CSIR CDRI, Lucknow. Dharmendra Kumar, Miranda House. Himanshi Sharma, Hesit. Monica Kumari, D. Crust, Haryana. Dr. Manju Shri Pawar, Paher, Udaipur. Santosh, Kumaon University, Nainital. Cherish Parashar, NIMR. Renuka Harit, ICMR, NIMR. Rakshit Ameta, Paher. So this is our way to inspire all those students who participated by presenting them oral and poster awards. Hope many of you will become future scientists. Let's carry the spirit of collaboration and discovery forward as we continue our scientific pursuits. All ends well when science prevails. We would now like to conclude the event over a cup of coffee and request everyone to kindly join for a group photo after the national anthem.
जनगण अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंग विंध्य हिमाचल यमुना गंगा उज्ज्वल जलधि तरंग तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Jai Hind. Group photo, please. I request all the speakers and all to come for a group photo.